Good morning, sir. Namaste.
Good morning, Prashant. How are you?
I'm good, sir. I'm good.
डिपार्टमेंटला बसलाय.
Department. Happy. GH Patel building.
हा हा हा.
Yes.
तू सकाळी लवकरच पोचून जातो म्हणा.
हा 9:30 पर्यंत येऊन जातो.
Good
तर
morning.
बोलिये सर नमस्कार. अमन सर.
Good morning. Good morning.
Good morning. Good morning.
Good morning, sir.
Good morning,
morning,
sir.
Prashant.
Good morning.
How
Good
are
morning.
you?
I'm good. I'm good.
Doing good? Doing fine?
Yes. So miss this opportunity to visit personally, right?
Next year.
Yes. Next year, definitely you come to Nashik.
Sure. No problem. Yes.
This year, Saranjit Singh was not well. He
Yeah.
was under some minor surgery.
Yes.
So he cancelled his plan and he is our brand ambassador. He comes every year here.
Yes. Actually, that is the attraction for everybody.
Ah,
Such a stalwart is coming regularly, and his name
therefore,
is connected with you, right?
therefore we took a decision to make it a virtual this time.
Right. No.
Next year, he has also promised he will come. So everybody will come.
Yes. So, what is current age he is running at? Almost 670?
He
No, I don't think so. He must be 66 around.
Okay, 66. Okay.
66. Yeah.
Mhm.
He must have superannuated last year,
Okay.
if
Yeah.
I'm not wrong.
Right.
Shall we start?
Yes.
Okay. So, good morning, one and all. Dear faculty members, distinguished speakers, researcher, respected delegates, and dear students. On the behalf of SNJB family, I, Dr. Aman Upaganlawar, extend my heartfelt welcome to each one of you for the Day 2 session of SNJB PharmaCon 2025. It is truly a privilege to have such an august gathering of academicians, professionals, and learners who share a common vision of advancing knowledge, innovation, and excellence in the field of pharmacy. Your presence enriches these events and inspires us to continue our journey of academic growth and community services. Yesterday, we witnessed three thought-provoking sessions. The first scientific session was delivered by Dr. Saranjit Singh, former director, NIPER Chandigarh. He shed light on the critical aspect of medicinal stability and its regulatory requirements. The second session was delivered by Dr. C.R. Patil, Professor R.C. Patel, Institute of Pharmaceutical Education and Research, Shirpur. He explored the fascinating world of artificial intelligence and mechanical machine learning in drug development and healthcare. The third talk by Dr. Prasad Thakur Desai, Chief Scientific Officer and Vice President, Indus Biotech Limited, Pune. He inspired us with his insights on endurance and resilience with fenugreek. Each of these sessions enrich our knowledge and set the stage for the exciting deliberations we will have today. With that momentum, let us move forward to our next distinguished speaker. Today, we are honored to welcome our first speaker, Dr. Prashant Murumkar. He is a young, dynamic, and enthusiastic researcher whose passion for innovation and commitment to advancing knowledge is truly commendable. Dr. Prashant Murumkar is a distinguished scientist and academician currently serving as an assistant professor at the Faculty of Pharmacy, the Maharaja Sayajirao University of Baroda, Gujarat. He holds pivotal leadership roles as the Associate Director of the Innovation and Startup Cells and Deputy Director of the Office of Alumni Affairs at the university. Dr. Prashant is a recipient of prestigious international ICMR Young Indian Biomedical Scientist Award in the year 2016 and 17. Recognizing his outstanding contribution to biomedical research, his global research footprint includes a tenure as a visiting scientist at Karolinska Institute, Sweden, the very institute that awards the Nobel Prize in Medicine. Additionally, in 2009, he was a visiting research scholar at the University of Utah, USA. A prolific scientist, Dr. Prashant has authored over 75 international publications, built up more than 2,250 citations with an H index of 25. His scholarly contribution extend to 3 books, 12 book chapters, and 10 Indian granted patents for research work on steroids, moieties for breast cancer treatment, and CB1 receptor antagonist for obesity management. Dr. Prashant's excellence in pharmaceutical sciences has been recognized with numerous honors, including the Young Research Award in 2024 by the MSU Pharma Alumni Association. Passionate about innovation, he continues to mentor PhD scholars in the design and discovery of novel medicinal compounds,
12:30.
shaping the future of pharmaceutical research. Recently, Dr. Prashant has been appointed to the role of scientific advisor by the Office of the Controller General of Patent, Design and Trademarks, Ministry of Commerce and Industry, Government of India. Today, he is going to discuss about nitrosamine impurities in pharmaceuticals, genotoxic risk, and ICH regulatory expectations. So, I welcome Dr. Prashant and over to you.
Thank you, Dr. Aman, for such a nice introduction. So, at the outset, I would like to thank Professor C.D. Upasani sir, who is our proud alumni of MS University. We always mention about his achievement, and everywhere we go, we give the name of some proud alumni, and he is one of them. Dr. Upasani is associated with the Chandwad College, and the same this conference every year. The name is with Professor Saranjit Singh, right? So this is one equation, and I have been following this conference since its inception. 1 or 2 times I could visit this campus. This time I was planning. I had a plan, but unfortunately I could not make it. But maybe next time we can have a physical interaction. So at the outset, again, I would like to thank principal, management, and all the teaching faculty members for inviting me in this prestigious conference and giving a space to interact with the participants. When I got a call that I have to deliver a lecture, so I was thinking that basically I'm a medicinal chemist. I must tell everybody, and I was thinking that this topic as well as conference related with the analysis, how I will give the justice to this conference as well as the audience. Then I thought that I should share some experience because, see, even drug regulatory agencies also they are in fact changing their views, and as and when there is a necessity, they come into picture and they put stringent regulatory aspects. So I thought that yes, I should talk about this aspect because since last few years, some of the pharma industries were approaching even medicinal chemists as well as analysts that uh, suddenly in the year 2018 there was incidents which was happened. As everybody knows that we have been taking medicine, particularly hypertensive medications, and there is a popular drug class which is a sartan class. You will find that there's a valsartan is there, losartan is there, and all this sartan class is there. And you will find that if you see the medicinal chemist point of view, you will find that there is a tetrazole ring which is present in this structure. This is a antihypertensive compound. And suddenly, what has happened that everything was doing nicely, and there was a news which was appeared that uh, the company from China who were actually involved in synthesizing this valsartan compound, FDA has asked that there is a certain cases of cancer which has been reported, and suddenly they found that there is a particular reason which is there that although there are certain medications which are having potential compounds in that structure which may prove nitrosamine impurity, but nobody was that much of serious. But when there was a case which was reported in the year 2018, then story started, like everybody was scratching, everybody was finding that whatever compounds are there, whether these compounds possess nitrosamine, whether this nitrosamine impurity is really harmful or not. Then investigation started, and then it came to know that yes, lot of medications which we have been consuming regularly, it contains the impurity called as nitrosamine, and there is a genotoxicity which is reported with that. And then FDA came with the resolution that we need to prove that whatever compound you are synthesizing, if there is a probability of having a chemical which is interacting with the solvent, or even I think most of these speakers, they must have talked about that there are certain aspects which are there while handling the medication, there is a problem with the impurities also, and there is a production of certain impurities. Although it is in trace amount, we need to prove that it is in the allowable region, and even there was not a standard benchmark which can be considered by the FDA. So that's why there was a, again you will find that there is an amendment which is going on. And I thought that yes, I should discuss this story with all the participants, that why there is a seriousness about the geno- genotoxicity which is caused by the certain popular medications which is having impurity like nitrosamine. So we will try to understand what is nitrosamine, why this nitrosamine impurity, and how many different type of impurities may happen there. And this is not like only medicinal chemist will understand. Definitely there will be a pharmacologist, there definitely there will be a analyst, and definitely those who are dealing with the pharmaceutical science, they should understand, and they should be understanding the concept of the impurity which is really threatening now because of cancer causing agents. And that's why actually we have chosen this aspect. So you will find that we will be talking about nitrosamine, we will be talking about genotoxicity. This is the second aspect, and third aspect is related with the ICH guidelines, because as I told you that even FDA is just recently, you can just google it out, you will find that there is amendment. Just last month, I was there in the Hyderabad to attend one of the conference which was organized by the uh, company called as LASA. In fact, European Federation as well as FDA has given authority to these certain companies that you come up with the softwares, and they have given approval now to this software that although you are proving with the experimentally, you need to prove with the theoretically also, that whatever drugs are there, you just put this drug into a software, and it will predict whether it is causing certain genotoxicity or not. So you will find that even FDA is relying on the private partners who are in position to give the information to the FDA. Thereby, FDA can give this regulation to the pharma industries. So as far as pharmacists are considered, we can generally produce certain evidences, and we can come up with the data. This data can be verified with the FDA, and FDA can come up with the revised regulation. That is what actually I have told you, that expectation from the ICH guidelines. So the title which I have mentioned is nitrosamine. I will be talking about guidelines, and I will be talking about various examples. That is what actually today I decided. So thank you once again for giving me opportunity to interact with all the participants with respect to this topic. So maybe just I'm sharing my screen. Let me know whether my screen is visible to everybody or not.
Not yet. Yeah, it it is now.
Okay. So we will be understanding why nitrosamine impurity and what are these. So those who are not having background of this, maybe so basically, you will find that there is a nitrosa functional groups which is bonded to amine. So this is what actually I have shown you here. So again, if you see the structure of amine, there is chances that you will find that if one hydrogen replaced of this nitrogen, we called as primary amine. If second is replaced, we called as secondary amine, and then third hydrogen is replaced, we call as tertiary amine. Right? Now you will find that this nitrogen is attached with the nitroso group, this functional group, and again this amine would be primary as well as secondary amine. So it was identified that this is the uh, initially identified, which is detected in the substances and drug product, which is called as impurities. So you will find that these are nothing but the organic compounds which are formed when nitrogen containing chemicals like nitrites and amines reacts with certain substances. These compounds actually possess potential risk of public health, and you will find that these reports have associated with nitrosamine impurities with cancer, liver damage, kidney damage, making regulation essential for public safety. And that is why actually everybody is talking about. Now there are small companies, they have been asked by the FDA, if you are in the manufacturing of certain drugs which is having amine like structure in that compound, and there is a chances that if you are using a solvent which is causing to have these nitrosamine impurities, you need to prove that whether there is a potential risk of this cancer causing agent or not. And that's why we are talking about this. So there is a N-nitroso dimethylamine, NDMA. Second impurity is N-nitroso diethylamine. Third one is N-nitroso N-methyl 4-amino butyric acid. Then another one is called as N-nitroso isopropyl ethylamine. And then another one is N-nitroso methyl phenylamine. So all these you will find that these are identified as detected in the drug substances and drug products. So this is nothing but the chemical structure of 7 potential nitrosamine impurities, which would be present in the active pharmaceutical ingredients as well as drug products. In fact, we can expect that in the API as well as in the final products uh, of the formulation, this type of impurities may arise. So this is what NDMA is there, N-nitroso dimethyl. You can see here common group is NO. Then you will find that here again dimethyl amine is there. Then another called as NDEA, nitroso diethylamine. You can see here this NO group is common, and again this amine structure will find that there is a diethylamine group is there. The next one is nitroso methyl phenylamine. This is what actually you will find that this nitro group is common, and then here methyl group is there, and another hydrogen is replaced with phenyl group. Then you will find that here there is a nitroso diisopropyl amine. Then nitroso isopropyl ethylamine group is there. Then nitroso dibutyramine is there. And then another last one is called as NMBA. This is what the structure of this compound is. You will find there is NO group is there, and there is a N-methyl 4-amino butyric acid is there. This type of impurities are there. Now, question is there how nitrosamine forms. So I'm not going in detail of this chemistry, but definitely there is a nitrites or other nitrogenous agents are present, and there is I told you that there is a secondary or tertiary amine combines under right conditions, and then there is a finally mutagenic carcinogen which is formed, which is called as N-nitrosamine. Now there will be different mechanisms for this. So as far as this in drug products are there is a nitrite formation is taking place. These nitrites form highly reactive nitrous anhydride under mildly acidic conditions. Then amine reactions are there. If nitrites reacts with secondary or tertiary amine present in the excipients also, that's why there is a chances of formation of this impurity. Then even we can consider there is enzyme pathways are also involved. You will find that there is a nitrates which is present in the excipients. It reacts with the enzymes whenever the drug is consumed. So that's why finally you will find that it interferes with the enzyme pathway, and thereby there might be a chances of forming impurity, and that impurity already established that there is a problem with this. And then even we can expect that there's a in the final product there is an N-nitrosamine form requiring the monitoring of the drug safety. Now, this is one aspect, right? So now there is the ICH guidelines which talks about primary genotoxicity. So that's why actually I have mentioned here. You will find that there is a importance of this ICH, particularly S2R1, and there is a ICH M7R2. These two guidelines are primary guidelines are important related with the genotoxicity. Now, what this ICH S2R1 talks about, it focuses on the testing and interpretation of genotoxicity data for drug substances. And then second one is called as ICS M7. It is ICS M7R2. It addresses the assessment and control of mutagenic impurity. Now, what is the objective of this ICS guideline, which is related with the genotoxicity? The main objective is to detect the compounds that induce genetic damage. That means there is a gene mutations or chromosomal damage is caused, and it access their potential to be human carcinogen or mutagens. Then there are standard test batteries are there. The guidelines recommend a standard 3-test battery to ensure comprehensive hazard identification. That there will be AMS test. I think most of the students they must be aware about that. A test for gene mutation in bacteria. Then another one is called as there is an in vitro test for chromosomal damage in mammalian cells. So, and then third one would be an in vitro test for genotoxicity. Basically, this test for chromosomal damage using rodent hematopoietic cell is mentioned there in particularly assays in the bone marrow or blood. So this is what actually it talks about a different type of recommended standard 3-test battery test to ensure comprehensive hazard identification. Now, what is the interpretation? Already I told you that the guidelines provide standard for follow-up testing and interpretation of positive in vitro and in vivo results, and it indicates how to assess non-relevant finding, or it determines if an in vitro positive test is not relevant in vivo due to factors like toxicokinetics. Uh, as far as this ICS M7 is considered, I have mentioned to you, is nothing but mutagenic impurity, and the ICS M7R2, it focuses specifically on the assessment and control of DNA reactive mutagenic impurity that may present in pharmaceutical products rather than in the drug substance itself. So we will be talking about not only the API but pharmaceutical products also. So you will find that there are certain principles which are involved in this. So there is a structure activity relationship. So that's why even FD has mandatory made it compulsory that now you come up with the guideline not only to prove with the experimentally proven experiments, but you can also have certain softwares which will predict on the basis of the training data set, which was already used to develop the models. So you'll find that there is a computational toxicology using QSR models is applied to predict if an impurity might be genotoxic or not. And then you will find that there is a threshold for of toxicological concern is there, which we call as TTC, for impurities with unknown genotoxic potential or those assumed to be genotoxic. So this TTC value is used to define an acceptable daily intake. So that's why if at all there is a impurity which is present in the final pharmaceutical product, then how much amount of impurity is allowed so that it will not cause cancer. So that's why, for example, it is 1.5 microgram per day is generally considered to be associated with an acceptable lifetime cancer risk. Then another principle is that risk management. So this guideline outlines a framework for risk assessment, then characterization, and what are the different mitigation strategies to control the this risk based on the safety and quick the quality of the risk assessment principles. So these guidelines work in concern with other ICS safety guidelines and quality. So that's why we call it as S series and Q series guideline to ensure the overall safety and quality of the products. So I wanted to just convey the message to the audience that why this nitrosamine impurity is important for us to understand, and why FDA is and also giving you certain guidelines. What is the objective of these guidelines? That is what actually I wanted to convey the message. Now you will find that it is proven record, and in fact the risk of colorectal cancer was also detected not only just recently, but it was reported in the year 2000 with this exposure of nitrite and nitroso compounds. So there was certain follow-up studies which was there, and then finally there was a conclusion that there is an association between intake of NDMA, that is what actually we have talked about, the kind of one impurity, and subsequent occurrence of the colorectal cancer. So you will find that there is an occurrence of colorectal cancer. It was observed to be associated with NDMA relative risk, that is 2.12. Then if you consider a smoked and salted fish intake of this, the intake is, this is what the consumption of the food which is generally taken by human being, it is 2.58, and you will find that there's a curd meat is there, it is 1.84. So you'll find that there is a relative risk which is observed with this colorectal. This is proven data. Now, why there was a global concern? So in the starting of my lecture, I told you that there was incidents which was happened in China, and that actually there was a press release on 13th July 2018 that European Medicine Agency, they reviewed medicines containing valsartan, because this valsartan was having the detection of N-nitroso dimethylamine in this medicine, and the company which was involved, again this was from China, it was Zhejiang Huang pharmaceutical company from China. So there was a recall of this product valsartan, and you can just see that everybody worldwide, this valsartan type of compounds has been taken by a lot of people in all the countries continuously. But because of this first report which was there, so that's why there was a sign of recalling of these products from dozens of European countries, not only European countries, but there are certain other countries like Hong Kong, Pakistan, Canada, and even from certain cases of US also, the product was withdrawn. And what was the condition? Condition was because of the discovery of this N-nitroso dimethylamine, which was detected in the sartan, which was used for controlling the blood pressure. And it was actually sparked global regulatory concerns and increased scrutiny. Now again, see, this is not the only salter content compounds. What I told you, that there is a probability those compounds which are having this amine-like structure, and you are using the reagents for the manufacturing, that means the way the compound has been manufactured, it will also affect the final products, because the small quantity of the nitrosamine might be present because of the manufacturing process. If at all compound contains this type of amine like compounds, and there is a different reagents which are using, which may cause to these nitrosamine, definitely there is a chances. Now how effectively you are using your analytical technique to detect this impurity, that was the major concern. So that's why this challenge was there, that nitrosamine in drug product remained a major source of worry for us. They were also not having certain data where to recall all the product, or whether we should recall only valsartan. But we'll find that not only valsartan but ciprofloxacin, we have been using the ciprofloxacin since long. Again, omeprazole like compound was also there. So a lot of compounds were reported to have this type of problem, and there was a challenge. So you will find that the simple I have taken this content from the FDA. You will find that in the year 2018, FDA Office of Generic Drug notified of NDMA in valsartan. Again, you will find that September, October, you will find that there was another molecule from the same category called as losartan, irbesartan. It was recalled because of the same problem called as another impurity, which is reported to be NDEA. Again, this is a type of one nitrosamine impurity was there. Again, you will find that not only valsartan, not only losartan, not only irbesartan, but there was a case with the losartan also, and that's why FDA again intervened. And again, in the month of February 2019, there was a detected impurity of NMBA, which is causing cancer. It was reported. That's why again the FDA has taken the stringent action. Not only stringent action, but they have decided that we should study all other compounds also. So now focus was not only just sartan. You will find that there's a ranitidine we have been using since long. You can see that again after 6 months of September 2019, there was a serious concern because of the NDMA impurity which was there in the ranitidine, and it was recalled because of the presence of this impurity called as NDMA, same impurity which was proven to be there in the valsartan drug. Again, month of, so you can see that now consequences are there, that everybody was aware, even FDA was aware, and they were asking all the compounds to be tested for this, those who are having this type of compounds where there is a chances to have nitrosamine impurity. Again, in December 2019, you will find that not only just ranitidine, but metformin compound also, and which has been used frequently by each and every patient. You will find that worldwide this metformin is prescribed. There was a problem which was reported in January 2020. You can see the nazatidine is there, which was recalled because of NDMA. Again, April 2020, you will find that ranitidine products were recalled because of the FDA's request. It was not proven, but they recalled that you just better get it checked, and that's why there was a recall of this ranitidine. Again, you will find that metformin extended release product recalled due to this NNDA. Then in August 2020, rifampicin. Can you imagine rifampicin? Rifampin reported to contain MNP. This is 1-methyl 4-nitrosopiperazine. This type of impurity is there. And then another compound is called as rifampantane is there, which is having this another impurity which is called as 1-cyclopentyl 4-nitrosopiperazine. This impurity was there. So you will find that there are certain these varenicline is there in 2021, which it was the tablets were recalled due to N-nitroso. Varenicline, this type of impurity was reported there. So this is how actually FDA was in action, and then So again, what the FDA did, there was a FDA nitrosamine task force was assigned, and they developed and published testing method for regulators and industry. Again, why I'm mentioning this, that even it was reported with the salted, it took another 1 or 2 years to take the decision about the other compounds. Thousands of compounds were there, and all these compounds were having this type of problem, but data was not sufficiently proven unless it was not tested for a particular action. So that's why it was like almost, even if you see today also, there is amended forms which are coming just in the April or June 2025. You will find that there was some amendment which was given, because even FDA doesn't have that much of strong data to make it in the regulatory form. So you will find that there was a task force which was assigned. Then it developed and published testing methods for regulators and industry, because unless you are not proving it, so that's why the testing methods were also assigned for that. You will find that the FDA has organized virtual public workshops where they will be talking. They talked about nitrosamine as inputs in the drug, the health risk assessment and mitigation, how to remove this type of problem, that was also discussed. And then FDA actually came with the guidance for the industry. There was a control of nitrosamine impurity in human drugs. It was published in the 2020. Then the regular meeting with other US government agencies, other stakeholders, and international regulators were there. Then you will find that there was certain actions to control that nitrosamine impurity, what should be done. So that's why mitigation, regulatory discretion, and recalls as and when it was required, they have implemented that. So you'll find that the guidance for the industry was applied. So the control of nitrosamine impurities in human drugs, it was the guidance was published. So potential sources of the nitrosamine impurities in active pharmaceutical ingredients (APIs) were defined. Then, as per these guidelines, you will find that there is a potential sources of nitrosamine impurities in drug products were also reported. And then they have recommend. There were certain recommendation to API manufacturers where the recommendation to drug product manufacturers were there, and there were recommendations, recommended timelines for the risk assessment, testing, and reporting was given under these guidelines by the FDA. So under this recommendation, what they have reported, that the assess the risk of nitrosamine impurities in APIs, not only APIs but marketed products of all the products, and product under approved and pending application. Those products were not given by the FDA approval, they were also tested for that. There was a guidance which was published. Then manufacturers do not need to submit risk assessment document to the agency, but they should retain this document so that they are available if requested. Because since this type of problems were reported, so that means a small groups who were actually involved in the manufacturing of this drug, they were asked to have these documents ready to be submitted to the FDA, which was related with the risk assessment which was given by the FDA. Then they conduct confirmatory testing when there is any risk for the presence of nitrosamine impurity, and report changes implemented to prevent or reduce nitrosamine impurities in APIs and drug products to FDA. So these are the recommendation. Now again, another question was there, that okay, it was proven that the certain API, certain final products were having these impurities, but what are the sources of nitrosamine impurities? So the main source was those companies who were actually manufacturing these compounds, having this certain type of compounds, so those compounds having this amine containing under acidic conditions, reaction conditions, if the compound is treated, there is a nitrite salts may form because of the nitrous acid, which can react with secondary, tertiary, or quaternary amines to form nitrosamine. This was the major source of the nitrosamine impurity. The API, then even detergents, intermediates, or starting material may contain secondary or tertiary amine functional groups. See, although not only just API which is having this amine, but those detergents as well as intermediates, as well as starting materials also. If it is having this primary, secondary, or tertiary amine functional group, then there was a chance that the nitrosamine impurities might be present in the final product. Then tertiary and quaternary amines may also be added intentionally as reagents or catalyst. Then recovered solvents. reagents, and catalysts may contain residual amines such as trimethylamine or diisopropyl ethylamine. If the recovery process involves quenching step, generally we talk this quenching step in the chemistry, where nitrous acid is used to decompose residual azides. Nitrosamine could form during the solvent recovery also. So it is important to understand that not only just the final product, how it is prepared, the starting material of the product, and even it was recorded that those vendors which were involved to supply the starting material, they were also asked to prove that your starting material having this type of impurity, and if this type of compound is used for the manufacturing of the final product, it is generating nitrosamine or not. If it is generating, then what is the impurity quantity which is getting generated, it was required to prove. Again, there were certain recommendations which were given to the API manufacturers. It was related with the optimizing the manufacturing process to minimize or prevent the formation of nitrosamine. So, as I told you, there are certain small groups who were actually synthesizing certain compounds and having this type of functional groups in the compound. So, they FDA regulatory bodies ask that you prove that whatever manufacturing process is there, can you replace certain starting material, can you replace certain solvents, so that there would be a minimum formation of the impurities are there. Even there were recommendations that you consider removing quenching step to avoid nitrosamine formation. Then audit supply chains and monitor of the at risk raw materials, starting material, and intermediates. Avoid cross contamination of the solvents, reagent, and catalyst. Then reprocess of rework API batches to control the level of nitrosamine impurities as provided in the ICS Q7, which we talk about pharmaceutical ingredient, which was given in the September 2016. And again, there was certain amendments for that. So for amending and controlling such operations. So now whatever guideline was there, depending on the output of the study or output of the reports which is provided by certain manufacturer, there were certain amendments which was carried out in the year 2024. Again, finally, because to this recommendation was given to the API manufacturers that you should develop a strategy to ensure that nitrosamine level remains within the acceptable intermediate region limit, allowable region. Again, not only just API manufacturer, but drug product manufacturers will also asked to follow certain recommendation. So they were asked to conduct risk assessment to determine the potential for nitrosamine impurities in drug product. Then test representative samples of incoming components, including lots of at-risk API priority use as required under different sections, and evaluate whether nitrites could be present during manufacturing process where at-risk APIs are used, and evaluate whether nitrosamine could form in a finished drug product or the drug product shelf life or not. So this again, it was not feasible for even FDA also, that what should be the acceptable intake for this. And then they collected all the data, and because you can see that there is a acceptable intake which is required on the basis of daily exposure to a compound that approximates 1 raised to almost 1 lakh cancer risks for 70 years of exposure. Again, there is a formula also. I have not included that how to calculate the acceptable intake, but you will find that by calculating this dose, the term acceptable intake is used in ICH M7's R1 guidelines to indicate the maximum limit for the impurity to be associated with the negligible risk of carcinogenicity or other toxic effect. If it is there below that recommended range, then it is okay. You can go ahead with the manufacturing, but it is beyond that, then you are not supposed to manufacture that. That was the major condition which was applied by the FDA. So FDA recommends the following acceptable intakes limits for the nitrosamine impurities: NDA, NMBA, NMP, and other impurities. Again, they have actually recommended that manufacturer use this AI acceptable intake when determining limits for the nitrosamine impurities. So you'll find out what was the AI limit. Again, find it. It is given in a nanogram per day. So if nitrosamine exposure is there for a particular person, healthy human being, below 96 gram nanogram per day is acceptable for a NDMA. That is what actually N-nitrosodimethylamine. you will find that there are other impurities are there. So it is allowable for 96 is for NMBA Ng per day, and the other impurities is 26.5. This is what actually allowable. This reach acceptable intake limit is mentioned by the FDA. Again, it is surprising to know that these nitrosamine impurities are there in the rifampin as well as rifampicin is there. So we'll find that again these drugs which we have been given to the patients regularly, there is a longer exposure is there, right? So we'll find that this nitrosamine levels exceeded AI for rifampin and trimethoprim, but both are used to treat tuberculosis. Although it was exceeding with the acceptable range, but it was given. So you will find that there is a 1.4 million deaths from TB in 2009, and 10 million people are suffering from that. So given the vital importance of this drug and the potential consequences of not treating TB, patients were advised to continue. So that's why I told you that FDA also is not in position to make certain decisions. So if we ban this drug, rifampin, what will happen? Because you will find that almost 1.4 million deaths are there because of the TB in the year 2019, which was reported. So the number is high. So that's why we cannot directly ask the patient to stop taking medication, because what will happen because of that? There will be a problem of the TB, and patient may die. So FDA has made it not compulsory, but they have given advice that because of this, they have advised continue taking the current medication and consult with their healthcare professionals about any concern. If at all there is a problem which is reported, then it will be taken care, but continue with the patient, the medication. That is what the message was given. Again, this was calculated, and interim AI is a short-term strategy to maintain patient access while process changes are instituted to remove or reduce nitrous formation. Again, meanwhile, what FDA has done that whatever manufacturing process is there, you just try to modify and find out whether you are really getting connected somewhere by using any starting material, you are using any reagents or not, where there is a chance that nitrosamine. Can you change this process or not? That is what FDA has asked to everybody. And then they have given flexibility when patient access to medicinally necessary drugs may be affected if all product is recalled. So it is not possible that it will be stopped. It will be recommended to stop taking the medications because of this problem. So again, if you see the doses which they have mentioned is that, so FDA for this type of drugs to help avoid shortage and ensure access to these medications, FDA does not intend to object to certain manufacturer temporarily distributing rifampin containing MNP below 5 parts per million, or other drugs below 20 ppm until they can reduce or eliminate their impurities. So they were asked to rework on the manufacturing process, and then you prove that whether it is below the mentioned content or not. And then a manufacturer should contact the Center for Drug Evaluation and Research drug shortage staff when the testing of rifampicin shows level of whatever acceptable range is there or not. So case to case basis also, they actually were dealing with the drugs. Now, let's consider the FDA has given the per day maximum allowable impurity is either 96 or 26. But if there are some multiple impurities which are present in the, because see, medications we are taking continuously, not only a single medication, there is a combination of medications are also there, and multiple drugs we are taking also. So above limits are acceptable if drug product contain a single nitrosamine. Whatever actually content I have mentioned to you is for a single nitrosamine impurity, but there might be a chance that if more than one nitrosamine impurity is detected, so then what should be the condition? So in that case, total quantity of nitrosamine impurity exceeds 26.5 nanogram per day, right? So based on the most potent maximum dose, the manufacturer should contact the agency. That's why they have mentioned to it. That means there is no clear indications also if it is more than that. Ultimately, what should be the condition if more than one nitrosamine impurity is there and it is exceeding the dose? Then depending on the situation, how important the drug is given, whether it is really required to be extending the life of the patient or curing the disease, what is important, accordingly the decision would be given. Now again, this is the third aspect which actually I wanted to tell you, that whether we are in position to detect this impurity or not. Yes, we are in position, but again, what about the traces of the impurity which is present? There are certain methods which we need to apply, and you will find that again there is a detection of these nitrosamine impurities in certain anti-TB drugs they have mentioned. So FDA has given the link, and this they have published the testing methods also to provide option for regulators as well as industry to detect the nitrosamine impurities in the anti-TB drugs. So they have asked that you should validate first, and then whatever resulting data is there, then as per this data, actually the results would be submitted to the regulatory agencies, and then regulatory agencies will take the decision about that. So they have mentioned the LCMS method for detection of impurities is given there. So there is a again not only just LCMS, but you will find that there is a HRMS method is also they have recommended. Now again, impurity would be identified by using certain analytical methods. So first we should know that what type of impurities they are, actually what type of conditions are there. Already we have talked about this type of condition. There are four different type of conditions. So accordingly, different stages also these methods can be implemented. So there will be nitrite salts, amine present, as well as acidic conditions. So these conditions also may result into a formation of this impurity. That's why, while considering the various uh, analytical methods, we should be knowing that at what stage actually we are withdrawing the sample and the method is used. So accordingly, there is a scope that the analytical person can develop different methods to identify the stresses of amount of this. Again, we need to consider these sources of contamination also. So whatever, as a manufacturer, if we consider, even if you have been asked to develop a method, you need to discuss about the sources of contaminations also, whether it is because of the presence of the vendor material. So we need to have a different methods to test for that materials. So it is contaminated with the starting material and raw materials from suppliers, including nitrite impurities in nitrate containing materials. Then if it is because of the recovered solvents, because see, nitrous acid used in the recovery process can form nitrosamine, and it actually entered in the recovered materials also. Then already we talked about quenching process, because there is an addition of nitrous acid during quenching when residual amines are present. And then process failures also, because there is inadequate control of the temperature, pH, reagent, and then the manufacturing of the even reagents also, what kind of reagents is used in the synthesis, that part is. So the acceptable intake we will be talking, we have already talked about, is nothing but whatever the FDA has given the limit. Below that it is there, then for all the method it is accepted, that it is recommended, that it can serve as the basis for understanding the nitrosamine impurities and ensuring the product safety. They will allow you to. Then there are certain case studies I wanted to discuss. Out of that, one is which is mentioned by the even FDA also. The capsule manufacturing consideration is there. Not only the product, but can you see here the kind of doses we are giving to the patient, not only because of the API, but because of the capsule, because of the starting material, there is a chance that these impurities might be generated. So there is a preventive strategies which is mentioned. The capsules are primarily made of gelatin and HPMC, and it contains various amino acids. So to prevent nitrosamine formation, actually FDA has again given guideline to the capsule manufacturers that avoid strong acidic condition. You are not supposed to use the strong acidic conditions. Control use of solvents carefully, minimize nitrosating agents, and implement rigorous testing protocols so that there will be a close examination and we can detect the method which are essential for identification of the nitrosamine impurities before releasing the capsules. Now coming to the what should be the actually our commitment to the safety. So our target is that there should be a zero nitrosamine impurity. So if we consider this capsule manufacturing, whatever hard gelatin capsule cells are there, then cellulose capsule cells are there, it should be free of nitrosamine impurity. Then there should be rigorous testing has to be there, comprehensive quality control, which ensures that there should not be a risk of nitrosamine presence. And finally, we should be giving safety as well as regulatory compliance. There, customer assurance should be there as far as this is there. Now, this aspect was already we have discussed. Now, how FDA is evolving themselves, that is what actually I wanted to tell you. So FDA has asked that you prove with the experimentally proven data that whatever allowable region is impurity is mentioned, that type of impurity you can prove, and then you submit this document to the FDA, and FDA will give you the approval for that, right? But again, they have also approached to other companies also that whatever data is there, this data you can use this data in the form of training set. Nowadays, everybody is talking about AI. Now there are different AI models are available. You must have seen that there is a search engines are available. Different AI models are available. Now those AI models which are giving 100% accurate results. Why they are giving 100% accurate prediction is there 100% because they are having the data set, the kind of data set which is giving you true results, right? So that means they are relying on the data set which is available. Same thing is available, applicable here, that whatever output of the result is there, that output result can be considered as a training set, and this training set can be used for those compounds which have not been yet synthesized, or already it is synthesized, still you can submit, and then you can submit that how this compound is manufactured, what is the starting material. So whatever protocol you are using to synthesize, that protocol has to be submitted. Then what will happen, the training set data set will be utilized to, on the basis of existing results, to predict whether the given compound is given compound is problematic or not, whether it is causing cancer, whether it is toxic or not, it will predict. So you will find that there is a LASA, the one organization is there which is UK based, and they have come up with different modules, and there is a even FD has asked nowadays that you get it approved with the experimental results, as well as you can get it approved with the prediction of the softwares also, and that is what actually both the solution is given by the LASA. So the company actually they have developed a software called as Direct, and then other software is Saraha Nexus. So Direct Nexus and Saraha Nexus both are operating differently, but what they are doing, I will briefly explain to you. These are the tools which are highly significant to the companies, because these tools comply with the ICS M7 guidelines. Now, why FD has asked this direct nexus or Sara nexus to use this? Because this ICS M7 guideline requires the use of two complementary in silico prediction methodologies. Now, what it does, it accesses the mutagenic impurities in pharmaceuticals. Right. So that means as ICS M7 guideline is also talking about the in silico as well as computational prediction methodologies. It also helps to reduce the animal testing, because if you are using computational tools to predict toxicity, companies can significantly reduce reliance on the costly and time consuming in vitro and in vivo animal testing, aligning with the principles of 3Rs: replacement, reduction, and refinement. So that's why actually these tools are highly significant. And then another important achievement would be that there will be a streamlining of the drug development, because these tools have ability to quickly identify and assess the potential toxicity early in the development of process, which helps to accelerate the timeline for bringing new safety drugs to the market. Now, whatever we have discussed, what will happen that if at all nowadays those who are actually interested to put get it approved, although it is off patented drug, but you need to, because most of the time what is happening right now, just yesterday, I have seen that there is a seraglutide is there, right? This anti-diabetic drug which has been given, it was there in the Times of India day before yesterday, that there is a Novo Nordisk was given a patent for this compound, anti-diabetic compound as well as anti-obesity compound, and this compound's patent is getting expired in the month of March 2026. Now, there are certain company like Dr. Reddy's, I have seen Sun Pharma is there, and there are other companies also there. What they are doing now, whatever manufacturing process of this anti-obesity drug is there, they have modified the manufacturing process, and they are coming with the drug, and now they are actually asking to do the marketing of the drug once the patent is that means after March 2026, right? So what they have done, the Sun Pharma as well as Dr. Reddy's, they must have not used the same manufacturing process of the patented compound. They have modified the manufacturing process. Now, because of this 2018 issue, because of this issue of this nitrosamine impurity, now everybody need to prove that whatever starting material they are using, everybody need to mention that what type of reaction conditions you are using, and you produce the data that whatever manufacturing process you are using, it is not generating the impurities. This is one aspect. Now this is not mandatory, but along with that there is another thing which is important, that you need to use certain softwares which has been mandated by FDA, that you put your compound in the software, mention this all protocol which you are using, and predict. If this software is predicting that whatever outcome of this result is that it does not prove to be a carcinogenic having impurity, then the FDA will accept your application to do the marketing or to give the approval for the marketing. That's why actually this is the important thing. That means you produce the data, and even whatever other people they have generated the data, use this data in the form of training set. And this, that's why actually this ICS M7 guideline is talking about computational approach. So there are two aspects, although there is a computational approach, but there are again sub approaches are there. One would be on the basis of experimentally proven data, and another would be on the basis of prediction, which would be done on the basis of training set. That is what actually it is mentioned in that. So this is what actually I wanted to talk about. Now there is a Lhasa company which talks about different modules. These modules are developed in line with the prediction of the toxicity. So there is a ICH guideline which talks about M7 guideline, and as per this, there is a direct nexus software is there. There is another software which is called as Sarha Nexus. So what is this direct nexus? It is an expert knowledge based software which can help you to meet ICS M7 by providing fast and accurate toxicity predictions. This is what actually this direct is done. And the SARA is nothing but it is a statistical based software which can help you to meet ICS M7 by identifying potentially toxic chemicals through the prediction of mutagenicity. This is two aspects which have been given. Uh, Now I have given you the example of these AI-based models, right? Same thing is done here. What they are doing, they are actually collecting the data of all the drug molecules, and they are actually defining the pharmacophore. What do you mean by pharmacophore? Pharmacophore is nothing but the minimum structural requirement which is shown to the biological activity. So likewise, there would be a toxicophore which could be identified. So whatever subpart of the structure is there, which is responsible to give the toxicity, so that will be identified, and they will be identifying the subpart of the structure, and they will report that whether it is causing the cancer, whether it is causing the impurities, causing the toxicity or not. That will be identified, and it will record in the form of values. This exercise would be done for all remaining type of compounds, and then they will be having a strong data set which will be utilized. So you will find that there is a access over 40 years of expert structure activity relationship knowledge for the mutagenicity endpoint that has been done there. So what they have done, they have considered all this data. This data was utilized to well established structure activity relationship knowledge for the mutagenicity endpoint, and then this SAR knowledge is derived from both public as well as proprietary data sources, and is regularly updated as new data becomes available. And that is the importance actually. So what they do, they retrieve negative prediction for bacterial in vitro mutagenicity. This is what actually it is important to understand. So there is a streamline. The expert review process is there with negative predictions. So there will be a positive as well as negative predictions would be there for bacterial in vitro mutagenicity data. So this direct nexus, what it does, it gives an interpretable method to determine the reliability of the negative predictions when assessing the impurities under ICS M7 guideline. Then even they have simplified the expert review for the submission under ICSM-7 guideline. So there is a transparent, scientifically robust prediction, which is supported by high quality curated data with extensive coverage of the chemical space, it permits rapid expert review. And even you will find that there is it can be used in tandem with the Saran Nexus And VETIC, there is another module which is called as VETIC to assign an ICS M7 classification for impurities of an active pharmaceutical ingredient. So what they do, so basically they are having updated data set. They are having retrieval of the negative prediction for bacterial in vitro mutagenicity data set, which is combined there. And not only just predicting it, but there will be expert review process would be there. They will help you to identify the results, and then finally they will give you the results in the form of the review experts. That is what actually this direct nexus does. Now there is another. Now, as I told you that FDA has made it mandatory that you need not to just there will be a validation of this method has to be there. So what they have done, they have asked to predict by using one software which is considered to be a direct nexus, which is considering the data of already existing, and then another there is a statistical based model is also there. They are asking you to validate the results, and that's why for the validation purpose they are asking you to predict by two different the in silico methods. And that's why there is a mandatory thing that not only just expert based software. Why it is called as expert based? Because you will find that whatever outcome is there, this outcome would be reviewed by the expert committee, and they will give you the suggestions whether the given compound is uh, giving certain toxicity or not. That part is important. And then another, the conclusion of this study would be supported with the help of Saran access, which is nothing but statistical based software which can help you to meet ICS M7 by it will help you to identify the potential toxic chemicals through the prediction of mutagenicities. That's why you will find that Saran access highlights are there. There is a prediction of the bacterial mutagenicities there. Here they will employ a unique machine learning methodology to predict the bacterial mutagenicity of a query compound. So what you need to do, you need to just submit the compound, and once this sub compound is submitted, this compound would be predicted the bacterial mutagenicity, and then you need to apply the Saran access as a part of MCS workflow is there. There will be a combining of the Saran access as well as direct nexus prediction. It provides you the means to meet the computational toxicological assessment, and then finally it will tell you whether the given structure would be acceptable for that impurity or not. And then there it will actually utilize the donated AMS mutagenicity data set. So there, whatever published data is there, that data would be covered. Right. So this is what actually I wanted to discuss with you. I will show you the software which has been mandated by the FDA. Yes, I will show you. Are you able to see my screen?
No.
Okay. Just
Nothing yet.
okay. Just 1 second. I'm now.
Yes.
Yeah. So this is what actually I wanted to tell you. So this the mandate has given by the FDA. Uh, this is the name of the company, Lasa Limited, and they have actually developed different tools. Out of that, two tools which I have mentioned to you is nothing but direct, and there is another Lasa is there. So So can you see here? There is a different solutions which have come up with. Nowadays, they have come up with the mutagenicity assessment, skin sensitization assessment, carcinogenicity assessment, genotoxicity, impurity evaluation, Non-genotoxic impurity assessment is there, safety profiling, drug discovery, impurity and detergent degradant control, then metabolite identification analysis, and then nitrosamine impurity risk assessment. So I was talking about genotoxic impurity as well as nitrosamine impurity risk assessment. Now you will find that the small process impurities and nitrosamine drug substance related impurities, which it is called as NDSRS in the marketed pharmaceuticals, actually they need to investigate into nitrosamine formation potential and their mutagenic and carcinogenic activity. So you will find that there is a solution which is provided by that. So you can just monitor the risk of formation and toxicological concern of nitrosamine impurities through the different tools. I have mentioned to use Sarahan access. Can you see here? Sarahan access is there and Derek, which is given mandated by the FDA. So they have come up with the Mirabils is there, Mirabil Zenith is there, and there is a Vitic is there. So their purposes are different, right? So even you can see that the Mirabils is there. This is nothing but risk assessment tool to assist with the management of your mutagenic impurity. It carry over risk for potentially mutagenic impurities in drug substance synthesis. Then there is a Zenith is there. You will find that it is related with the degradation prediction quickly for a variety of post degradation conditions in the analysis, because generally we carry out this post degradation product. So if you give the different conditions, it will predict the nitrosetting pathways. Then you will find that there will be a, you can take the decision making around the experiments and excipient selection. That part also can be taken with the help of this Zenith. And then this is what actually I was talking about. Can you see the screen?
Yes.
Yes. So this is what actually nowadays everybody is talking about Saran access as well as Dere access, and not only just prediction of the determination of the impurities experimentally, but you need to prove with the uh, prediction by using the software. It is mandatory. You need to submit the data of this prediction. That is what actually all the pharmaceutical companies, either they are getting it done on their own or they are actually getting it done from through different agencies, and they're asking. So this is non-profit organization which is called as LASA. They are actually having experts people, and they are actually updating the data regularly. That is what actually I told you that just recently I was there in Hyderabad because they conducted one workshop and I was there, and that's how actually I came into in detail analysis of the compound. We have been using this since last few years, and the results are excellent for that. That even for giving just starting material, we are giving the reaction conditions, we are giving force degradation conditions. So it is very much possible that you can come up with the possible nitrosamine impurities, whether the given compound would be given, and by using this input you can just change your reaction conditions, and finally whatever safety parameters we are able to establish it, that would be helpful to us, so that we can go ahead with the synthesis part that can be done, right? So you can see here, direct nexus actually determines the level of toxicological risk posed during the direct nexus. It determines this. So again, there is a structure activity relationship data which has been considered there, not only for nitrosamine genotoxicity but carcinogenicity is there. So there is the results are given in the form of alerts. So what this alerts means, the alerts mean that whether the particular structure, part of the structure is responsible to have a carcinogenic potency or not. So that alerts would be given. And these alerts are nothing but the predictions, right? So it is also called as assessment of the regulatory carcinogenic potency categorization approach, CPCA, for nitrosamines. And it even compares the EMS mutagenicity using this Saran access data. Then there is a, as I told you that there, it is just not the prediction, but there is an expert comments would be given. Simple one-click method will give you the prediction made by the direct access as well as Saran access to provide an overall ICH-MC1 prediction alongside the semi-automated expert review. Argumented aim to make expert review more effective and efficient. That is what actually they are claiming, and that it is really useful to make the conclusion, right? And then there is a alerts, they are fired when compound is in the cohort of the concern, right? So this is the flag to alert the reviewer when the compound is expected to be in the cohort of the concern. Is included within the ICF's M7 prediction or not. So that part can be done. So they are now coming with the other softwares also. VTIC is there, retrieve in vitro genotoxicity study, which is the records of the nitrosamine. And then they can collaborate with the industry expert through data sharing also, and they will give you the expert opinion that can be done. So this is structure searchable toxicity database. VTIC is there, and then there is an information management system is there where the carcinogenicity studies are recorded, as well as nitrosamine data is also considered there. So these people, they will help you by using this VTIC to retrieve the existing data available for your compound, whatever compound you are, because always it is not possible for the industry that to talk about the novel compound. But because of this concern of 2018 issue, now each and every company need to actually go through this nitrosamine impurity, because directly or indirectly they may be in contact with the, if amine containing compound is there and there is a formation of nitrosamine is there or not. So that's why everybody is talking about this softwares also. So there are different databases are there. So right now, just I wanted to talk about these aspects with you all. So this is what actually I wanted to convey a message to everybody. If any question is there, please.
Okay, thank you, Dr. Prashant Murumkar. Now I request audience, this session is open for question answer. If you have any doubt, you can ask directly to Dr. Prashant. Yes, from students.
Dr. Prashant,
Hmm?
I have one doubt. Whether
Yes,
these impurities are self-generated or they are chemically generated.
they are chemically generated, sir. Uh,
During process of synthesis of that compound, these impurities are generated.
So there are two aspects. One is called as if we are using any compound which is having amine-like structure, right? And if you are using certain reagents which will call nitrosamine, right? So what will happen? This is a regular process which we carry out for the manufacturing of this process, right? So that means there is 100% chances of nitrosamine is there. But thing was there that FDA was also not sure whether it is causing genotoxicity or not. The limit was not detected. So it was there. Even thousands of compounds are there which is having nitrosamine. But nowadays, why? Because of this case of 2018, certain Devon, there is a synthesis of tetrazole is there. If you see the structure, and tetrazole synthesis requires this type of reagents, where there is a nitrosamine impurity is there. This is one aspect. And there is another aspect that intentionally you have not added it, but whatever starting material you are using, that might be having nitrosamine impurity, and it was not detected by the analysis, right? Or we were not incapable to identify nitrosamine. And for the starting material, because you are not directly using that compound, but nitrosamine is present in the starting material itself, which was not detected, and now it will be there in the product which was not removed. So that is the case. So that's why it is not always because of the intermediate is there. It might be there present in the starting material itself also.
Now is there any process control has been designed
Yes.
control these nitrosamine?
Yes, there is a process design, means there is an alternative route of synthesis where you need to use alternative for the given compound. If at all the vendor is found to be having nitrosamine impurity of that compound, you just change the vendor and you find out that if you are having analytical powers where it would be detected in the given limit, right? And so what you can do, you can just simply change the vendor. You get the another company's starting material, check it, and if it does not having nitrosamine impurity, you can use it. So this is one aspect. Another aspect is that finally you need to, whatever compound is getting synthesized, you need to work on reduction of these impurities. If you are able to prove that, then you can you can be allowed to market your drug. Otherwise it's difficult.
So is there any method available?
There is a method. Yes, there is a even HPLC method as well as LCMS methods are there, which actually has been developed by different pharma companies, where a small quantity of the nitrosamine can be identified. Yes, it is.
Okay, this
Yes.
is one thing. And is there any method developed to minimize these impurities?
Minimize, yes, there is a again, it is case to case study is there. Like suppose I can give you the omeprazole mutagenicity complex is there, right? So there are certain tests are there, which we talk about. Many standard tests are there, like AMS test is there. It shows no direct bacterial mutagenicity is there, right? But sometimes you will find that in vitro as well as in vivo studies suggest that there is a mild genotoxicity effect is there, where there is a DNA damage is there, chromosome issues are there in mammalian cells, possibly which is linked to its there is a sulfonamide metabolite is there, or increased gastric pH, allowing more reactive compounds are there. So you will find that because of these studies, there are certain tests which you can apply, and it will predict whether the given compound is having nitrosamine impurity or not.
Okay, thank you.
Yes.
Dr. Prashant,
Yes.
okay, you have highlighted many important aspects of nitrosamine. One question is there. As you said that whatever example you have quoted, all these drugs are used for chronic treatment of disease, right?
Hmm.
So if it is a chronic treatment, that means the exposure of nitrosamine impurities are for long period,
Hmm.
and that will causes the genotoxicity.
Yes.
So, if the drug is very rarely used, in that case, what are the chances of this genotoxicity?
Now FDA has clearly given the guideline that there is a allowable limit is mentioned there. Now
Okay.
again, what has happened that those drugs which we have been given to the patient since last few years or like decades, right?
Hmm.
For that, it is very easy to give the data. There is no problem. But now there are certain drugs which are there in the pipeline. So that's why they ask you on the basis of existing data. They have made the limit. Now, if it is below that limit, they are allowing you. There is no problem.
Yeah, it can be answered in one statement that prevention is better than cure.
Yes.
You
Yes.
don't cater medicines with impurities, nitrosamine impurities. Better to check it
Yes.
ahead of introducing it in the market than
Yes.
to avoid mutagenicity and oncogenicity.
Now, again, now it would be specific. Now, if you are talking about life-saving drug, where if you are not giving this drug, patient will die, right? So
Yeah.
better you give it, because there will be there will be a prolonged effect, and then that effect will be secondary. So
Yeah.
you need to take a decision accordingly. So that's why you will find that rifampicin, you will find that FDA has not taken action.
Hmm.
It is actually they have decided, you talk to the physician.
Yeah.
Physician will take the decision. Life is important, or this
Yeah.
mutagenicity is important or not,
Right. Right.
right?
Same
So
happen in case of cancer
yes,
treatment.
yes. So now my question is here again to the audience as well as to all the panel members, that see, we are frequently taking all these medications without concern, right?
Then.
But now there is a concern. Certain derivatives, I think we are asking our colleagues also, because those who are in 40, 45, they are, which they are taking the medication. There is
Hmm.
amlodipine, telmisartan is there. Telmisartan still we are taking. Now we are not worried about the regulation which has been given. Because are you asking to any company that whether there is a allowable nitrosamine impurities there or not? We are not talking still up. That means even FDA is not clear up to 2000. Just recently, I have seen that there is an amendment which has been made. Because if you suddenly stop asking the all manufacturers to stop manufacturing these certain derivatives, there is no alternative medications, right? So that's why I think still there is no 100% foolproof rules which has been made by the FDA, and we should be aware about these things. Because see, now what we should do, if we know the clear method to identify nitrosamine, let's check for those medication which we are taking right now, and find it whether it is allowable region or not. If it is allowable, then it's okay. If it is not, then we need to find out something else. You just test it and take the medication alternative of that molecule. Again, company to company, it is going to change. If you are taking Dr. Reddy's valsartan, if you are taking Cipla's valsartan, if you are taking Glenmark's valsartan, you should ask the data and then you start taking the medication. That is what major concern is there, right? And we are not talking about this part because we don't have anything in our hand to stop or to take the medication. There is
Yes.
no data available.
Right. Another thing is that the same regulations are applicable to cosmetics also, because many cosmetics also containing these nitrosamine impurities.
Yes, but actually, unless they are not penetrated into a main blood stream, there is no
Okay.
problem. Yeah, because if it is getting absorbed, and
Hmm.
then definitely that concern would be there. But see, we can ask to avoid using this cosmetics if there is
No,
a
no.
problem.
Prashant, I think it is said
Hmm.
that whatever you apply on skin,
Yes,
and
yes,
it is
it
bound
is.
to absorb.
Yeah, so that's why directly it can be like you stop the this cosmetics. That would be the concern, right? That would be the given. But unless it is not proven that it is getting absorbed and then it is because of the impurity that problem is there, nobody is going to take the action. But immediately, yes, we can ask that you stop because it is not given for any therapeutic activity.
And one thing is very much accepted nowadays.
Mhm.
Everybody is agreed
Yes,
that if nitrosamine-like impurities, they can cause, they are carcinogenic, they are dangerous. So at any concentration, in any limit, they will be showing their effect in due course of time.
yes, yes, absolutely right.
If the quantity is very less, then the effect will be there after 15 or 20 years. If
Yes, yes,
quantity is more, you may see within 2 years.
yes.
It will be there, but nitrosamine will not, uh,
Eliminated,
you just
eliminated.
can't avoid the deleterious effects of nitrosamine in due course of time. So
Yes,
it is always advisable to avoid that. That is
yes.
the best thing what we can do.
Now, but again, sir, question is that for antihypertensive medication, what would be the solution then? So, because
Ah,
it was the
but
safest molecule, right? Sartan was the safest molecule, valsartan, irbesartan. Nowadays, I think everybody is taking that medication. Still, it
hey,
is taken.
the industries will find their way
Yes.
to.
And so that's why, sir, I was surprised to see there were certain industry which was located in Vapi, in Nandesari, nearby Baroda, who were involved in the synthesis of this API. And suddenly they are asking that, can you help us to prove the data that whatever manufacturing process is there, it doesn't have that much of. So that's why I was surprised. And then I came to know that, yes, this is because of the stringent rules, which actually not yet made by the FDA, but it is going to happen. Otherwise, they
And
will be stopped. Whatever export is there, it will be stopped.
we should give credit to FDA also. They
Yes,
come
yes.
up with this strict guideline and to have a concerning role in public safety.
Yes. So nowadays, see what is happening right now. The API manufacturers who are located in a rural area also, they are planning to export their product to the manufacturers of the drug manufacturers. Right now, manufacturers are now so alert that they are asking you that you prove this, then only I will give the order to you. Otherwise, there will be another people who will be having certificate of this, and then the order will be shifted to somebody else. So that's why now everybody is alert.
This is a great opportunity for all the medicinal chemists if
Even
they
for
can.
analyst also.
Analyst.
No, no, chemist, if they can find a strong
Method.
way out
Yes,
to
yes,
Reduce.
reduce or to remove the nitrosamine impurities from the API,
yes,
that company will get a tremendous business.
yes, yes, yes. Absolutely right.
So
So
all medicinal chemists should
Yeah, this is only possible overseas because most of them dare to fund the medicinal chemist. And on this version, I would say that it requires lots and lots of instrumentation and heavy equipments like
doctor
that of LCMS to
medicinal
detect
chemists
impurity.
like medicinal chemists like you and Murumkar should come up with best possible alternative way to remove all the impurities of nitrosamine from a compound. If you are succeeded in laboratory scale, it can be scale up to the industry scale. There is no
yes,
problem.
yes, and even I agree that if we would be able to establish the method of detection also,
Hmm.
even method of preparation of impurity also, that would be a big achievement, right?
Yes.
Because that is also because the reference standard is also important. It is too costly. So yeah,
I agree. Detection will prevent the future loss.
yes,
But
yes,
but if you can remove the impurity and make the compound safe of nitrosamine,
yes,
that will be of huge interest and huge business. I am finding in that particular process.
yes, 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 absolutely right. And I think pharmacologist, because I think even FDA is also not allowing you to use the animals, right? So they are
Yes.
asking pharmacologist, you use the softwares which is having trained data and you predict the activity. Even they are asking to pharmacologist also not to use not to use animals.
I agree. Why they are doing that? It is for learning process. But when it comes to actual drugs, pharmaceuticals, and chemicals, we have to use animal. There is no restriction on use of animal.
Yes. Mhm.
They are trying to reduce the use of animal.
Yes, yes,
Thank
absolutely
you.
right.
Thank you.
Absolutely right. Mhm. Yes.
Okay, so thank you, Dr. Prashant Murumkar, sir. You have highlighted many aspects of nitrosamine, right from the basic to the genotoxicity, and what are the role and responsibilities of the FDA, how they are working in this matter, and the software which are available. Really, and definitely, we will try to use this software before going for any type of the screening. And thank you once again for sparing your time with us and sharing your knowledge with our audience. Now, the next speaker is Dr. Vishal Zamade. Now, I request Dr. Hitesh Share to take over the.
Yes, sir. Thank you, sir.
Dr.
Thank you
Zamade
very
is
much.
there?
Yes, sir.
Thank you
Thank
very
you,
much.
sir. Good afternoon, all. Am I audible? Okay.
Yes.
Respected principal, sir, esteemed resource person, distinguished guest, dear faculty members, researchers, and dear student participants. A very good afternoon to all. I welcome you all for the day second of SNJB Pharmacon 25. Myself, Dr. Hitesh Shahare, Associate Professor, Department of Pharmaceutical Chemistry. On behalf of SNJB Shriman Sridhar Jain College of Pharmacy, Chandwad, I am here to introduce today's eminent resource person of second session of SNJB Pharmacon 25. It is truly a moment of pride and inspiration for us to welcome whose academic journey beautifully exemplifies dedication, innovation, and excellence in pharmaceutical sciences, that is Dr. Vishal Prakash Jhamre. Dr. Jhamre sir is currently serving as professor and head of the Department of Pharmaceutical Chemistry at Srimati Kashibai Navle College of Pharmacy, Pune, and brings with him more than 20 years of rich academic, research, and administrative experience. Sir received his PhD from the Maharaja Sayajirao University of Baroda in 2012. Sir completed his post-graduation from Shivaji University, Kolhapur, and graduation from SPPU, Pune. Sir is GATE qualified scholar and honored with the National Doctoral Fellowship by AICT, that reflecting his strong research foundation. His research expertise lies in medicinal chemistry, computer-aided drug design, QSAR, and bioanalytical method development. He has an impressive research profile with over 400 citations and H-index of 11 and publications in high-impact international journals such as RSC Advances, ACS, Sustainable Chemistry and Engineering, Molecular Diversity, Journal of Chemical Information and Modeling. Sir published national books and several chapters in international books, and with this, many more. One of his research article has also been recognized as one of the highest downloaded paper by elsewhere, highlighting its global relevance. Sir has attended and presented the number of research papers at national and international conferences like Tetrahedron Symposium organized in China and USA. Sir is recognized as PG and PhD guide of Savitribai Phule Pune University. As a mentor and guide, Sir has successfully supervised over 40 MPharm dissertations and currently 2 PhD scholars are under guidance, shaping the career of numerous students and researchers. Sir has received research grant from NDF, AICT New Delhi, and also from SPPU Pune. In addition to his academic achievement, received several prestigious awards, including the Best Teacher Award, and has actively contributed as resource person, conference organizer, scientific committee member, and faculty development expert at national and international platform. Today, we are truly fortunate to have such an eminent scholar and passionate educator among us who will be sharing his valuable insight and experiences on topic entitled Navigating Regulatory Landscape and AI Trends for Drug Stability Studies, which is highly beneficial for all participants. On behalf of the SNJB's organizing committee, with great respect and warm applause, I request you all participants to welcome Dr. Vishal Prakash Jhamre sir. Sir, we are deeply honored by your presence and look forward to an enlightening and motivating session. Sir, please. Thank you.
That was very wonderful introduction. Thank you very much, sir, for this nice introduction. At the outset, am I audible?
Yes, sir, you are audible.
Okay. Thank you so much. So at the outset, I would like to thank the convener of this workshop, Dr. Professor Upasani sir, and also coordinator of this particular event, and our sir, Dr. Aman Ganlawar. And I also joined this session just before 5 minutes, and I could see one of my senior, Dr. Prashant, was presenting, and it was glad to listen to him again after so long time. So it was a wonderful moment for me to listen to him. And yes, in fact, I could see that the discussion was very in depth about the nitrosamine impurity. So this is a very technical talk which has been delivered by Dr. Prashant. So as far as my talk is concerned, this talk is more basic, and we will be focusing on more how this regulations in the drug stability came forward, how the evaluation process had been taken place. So basically, we will be looking at that. So entire path of evolution of regulatory framework that we will be going through. And secondly, this is very initial part, and then lastly I will be covering with what AI tools we can use and whether it is acceptable to FDA, if we predict the expiration period by using AI tools, whether it is acceptable or not. That of course we will be having on touch upon. So let me share with my presentation. Is it visible, sir?
Yes, sir.
Just a minute.
Yeah, it is fine.
Yeah, so it was a wonderful introduction given by the sir, but there is little change in the introduction because recently I have switched over to the another college, which is a private university, Pimpri Chinchwad University, School of Pharmacy. I'm working as associate professor there in pharmaceutical chemistry. So this is the small change I would like to tell to all the audience. Let us start with this presentation. Now, as far as when we talk about regulatory uh, framework, we always wanted to discuss about the quality attributes of drug substances and drug products. without considering drugs quality attributes of drug substances, we cannot think over how we should go for a protocol designing for stability studies. So as we all know that quality, safety and effectiveness, all these are the quality, important attributes for the drug substance. It may be sometimes physical, chemical, sometimes biological, or even microbiological properties with respect to purity, identity, its potency, and in formulation kind of words, if we say that particle size, polymorphism, the moisture content, all these parameters are to be considered while we talk about the quality attributes of drug substances as well as drug products. So here we all know that several types of drugs are there. We categorize those drugs in small molecule. Either they may be natural or synthetic. Second, they are biologicals. We call it as large molecules, and some biotechnologically derived products. So these drug molecules have to have some quality attributes while designing the protocol for stability. And when these compliances, if we want to comply these quality attributes, the process, or it should have a very simple and comprehensive and feasible processes, right? So that we can come up with the stability profile of that particular compound. Now this was a requirement for establishing the expiration period of the drug products or drug substances. Now when now let us see the historical aspect. Now what happened actually, the World War, till World War II, there was no such guidelines available to the entire pharma market for the testing the quality. Now forget about the stability, it was not established for testing the quality also. But later on, after the World War, the active pharmaceutical ingredient was tested for its quality by assaying it. That means just simple one assay, and whether it is pure or not. That's it. That was the only criteria for defining the quality of the drug substances. But what happened later on, and in fact, in before World War, the scientific understanding surrounding the drug stability and the minimal compared and modern standards of that drug substances was not established. So basically, during the World War or before the World War, there was lack of information about the regulation. And in that area, in that era, basically the reliance, basically the as far as the quality is concerned, more often it was relied on the pharmacopoeia. And that time pharmacopoeia was the established one, and that London pharmacopoeia was to, is to be referred, was to be referred every time by every pharma industry. Second thing, why this stability studies framework or regulatory framework is not there, because earlier in before the pre-World War era, most of the drugs were naturally origin. Like for example, they were isolated from opium, or sometimes it is isolated from coca plant. So these naturally occurring drugs uh, may not be having the stability issue. That time it was not established. So basically, when this stability comes into the picture, these products, which was isolated from the natural plant or source, they when they come to for packing, at that time stability came into the picture, right? So this was the first moment when the people in the pharma industry, scientists in the pharma industry, recognized that there has to have some stability concern for the drug molecules. Now, after World War, then the revolution had happened in the analytical instrumentation, because of the advent of the instrumentation technology as well as computer. And because of that, all efforts of scientists and engineers, the instrumentation came up. And as a result, what happened is the we could come up with the technology, some softwares or instruments which can help us in detecting the lower minimum concentration of the drugs, as well as the resolution of the complex samples which are having n number of compounds in a mixture, so that resolution can be enabled to have a resolved compound using those instruments. So because of this revolution, what happened actually, the pharma market started growing because of the consideration of the regulatory framework. Now, since the minimum lowest concentration can be determined by some techniques, so because of that, the pharma company have realized that there has to have some uh, regulation framework for this. And because of that regulatory framework, the pharmaceutical market automatically started lifting up towards the upside. So this was the need. I mean, this was the point where it was realized that there has to have some guidelines. Later on, in earlier days, if we talk about the earlier days about stability, in 1970, first time it was observed that unstable compounds are not maintain their quality attributes if they are storage for a longer period of time. So this was observed, quality attributes have started hampering because of the longer storage. So by keeping this in mind, in 1975, the USP came up with the drug expiration dating period. So this is the 1975, first time USP introduced it. And then afterward, expiration period was started. I mean, this guideline was started by implementing by all the pharmaceutical companies. Now, subsequently, for having this practice of expiration dating period for a few years, then observing this data, it was realized that there has to have some stability guidelines because we are getting some inputs from the drug expiration period. This was thought by the US FDA, right? So this in 1984, the first stability guideline was introduced. Subsequently, in 1987, US FDA published some statistical designs as applicable for stability studies to establish the expiry period. In fact, this rather than specific requirements, US FDA introduced guidelines on general principle of process of validation, rather, right? So initially they have come up with the validation process guidelines, and then they have introduced the specific statistical design, because and in that era, every pharma industry used to apply different techniques to the stability data, statistical techniques. So to make it on a common platform, these US FDA have come up with the stability guidelines for establishing the appropriate expiration period. Now, after having following these all guidelines which are introduced by ICH as well as US FDA, every pharma industry used to report stability studies in different formats. So it was very difficult for to analyze what stability studies are. How they are to be judged and how we should proceed further for a having the just a minute, madam. After few minutes. Sorry, it was some guest arrived in between. Okay, so it was. Am I audible?
Yes, sir. Yes, sir.
Yes. Okay. Okay. So we were discussing about the discrete data which was submitted to the bodies, and every pharma industry reported the stability data in a different forms. Therefore, the FDA come up with the guidelines on submission of the stability information for IND as well as NDA. So this is the beginning of actual practice of stability studies and guidelines, basically. Later on, having incorporated all these guidelines for IND and NDAs, 1993, first time the ICH, ICH guidelines have been introduced, and they are named as Q1A. As we all know that we are all following those guidelines, Q1A guidelines on stability. And then later on, 2003, Q1A guidelines, then QC5 guidelines was introduced. The Q1B photo stability guidelines, Q1C new dosage form guidelines, Q1D bracketing and matrixing guidelines, then evaluation of data Q1E and Q1F, that is for stability testing from climate 3 and 4 region. So these are all guidelines which are introduced over the period of time. Now what happened, the recent happening is now the Q1 guidelines. Now these Q1 guidelines are now in public consultation. Recently in September, there was a conference, RAPS Convergence 2025 conference held in North Carolina in United States, where the Mohammad Gaurab, you can see in the picture, who is the Vice President of Paraxel, he introduced that ICH has come up with a new guideline, that is Q1 guideline. And he designated this guideline as one-stop shop for stability testing. That means in a one document, every the analyst may get every aspect or criteria of the stability. Every corner of the stability is covered in this particular guideline. It is almost 108 page guideline. So what it includes, basically, this is a new guideline which is open for public consultation now, and scientists, analysts all over the world, they are making comments on it for even making it better. So what it includes, basically new information on in-use studies. Basically, these guidelines are more emphasized on the quality term. So in-use quality studies are more emphasized here. Then short-term stability studies, process and holdings. Yes, the process and holdings times are also considered. For example, if one intermediate is there, and if intermediate is being for make process of intermediate, if there is any instrument being used, so the contact period of that intermediate with that instrument, so how much it should hold, so that every aspects of the quality is included. Secondly, adjunct and reference standard studies, product life cycles studies. So these all are there for in the new this thing. Later on, after that, new part is information on how to address new technologies such as cell and gene therapy. So this is newly incorporated stability consideration for reference standard as well as novel excipient. So this is a complete revolution of the Q1A, and it is a Q1 guideline will be shortly introducing in the practice. So basically, if we go through this ICH Q1 guideline, there are three major types of stability studies are considered in the guidelines. First one is development studies under stress as well as force condition. So two conditions they have maintained. It was also earlier. Stress, the stress here emphasized, they want to emphasize stress as a condition that are not designed to degrade the sample, right? So we are not expecting this degradation product, but they are detected. For that condition, for that purpose, the stress studies can be carried out. And force condition is, it do contain condition that are intended to degrade the product. That means if we are knowing the chemical structure, then based on the chemical structure, what are the likely to be possibilities for having degradation product. So these studies are included. Secondly, formal studies. Formal studies are actually primary ongoing as well as product life cycle studies. Primary studies are basically all our these primary studies, what quality of drug substances as well as the environmental factors. Yes, in primary studies, environmental factors like temperature, humidity, and all are considered. And product life cycle studies, it is aligned with the ICH Q12 guidelines. So stability studies into the actually all holistically, the stability studies are considered in ICH Q12 guidelines. So these are addressed in the ICH Q1 guidelines. Thirdly, the supportive study, that means it is also earlier present in the guidelines, photostability in use short terms as well as excursion studies. So all these are new three aspects which are there in the ICH Q1 guidelines. Now, for designing a protocol for a formal stability studies, all critical attributes that we have saw in the earlier slides, those are to be considered, that are going to be changed over the period of time. So all possible quality attributes we need to consider while addressing or while designing the protocol for formal stability studies. And that protocol would become a most standard protocol to follow for the drug substances as well as the drug product. Now, this is the guideline which I was talking about, the Q1 guideline. So it is it is included in 11th April 2025, and these 3 points are new to the guideline. The bracketing and matrixing, these are the techniques which are used for reduced stability studies. The major challenge of stability studies is that it requires a maximum period of span to get the results. So these are the some strategies, bracketing and matrixing strategies. We can have a reduced number of stability studies, and then we can extrapolate to the what could be the possible stability of the product. These are the techniques for that. Then stability modeling is also there, and then advanced therapy medicinal product. That means these covers all cell therapy, gene therapy, and all. So this is all new to the guidelines. Basically, the stability modeling, the second part of this drug substance, is that it is nothing but your accelerated studies, like if temperature, humidity. We accelerate the conditions, and using all the results, we apply some statistical techniques, tools, sometimes the regression equation and all, and then we can predict the shelf life, right? So these are included in the stability modeling. So this is a new content. Basically, these are annexes, new annexes to the Q1 guidelines. Now, this is, I've included one bracketing examples. These bracketing examples are what? They are using the extremes of the conditions. Now, we can see here, this is the strength of a drug substances. These are the second row is column, sorry, second row is batch, and then these are container size. So extreme certain design factors are considered. For example, if we talk about 15 ml, then for 15 ml, 50 milligram the first extreme and 100 milligram second extreme. So only three, the 50 milligram 3 batches and 100 milligram 3 batches. Same for 500 ml, 100 ml. So extremes, if we say about dose wise extreme, the lowest extreme is 50 and highest extreme is 100. So this is the example. This is how the studies are planned, and they have made this example available in the guidelines. So yes, having said that, there are some certain challenges in stability studies, and it might be discussed in earlier sessions also. Say for example, as just now I said, that long duration and resource intensive accelerated studies are there. So time period is the major challenge here. Then the second one is high variability from batch to batch manufacturing, as well as the excipient manufacturing. Third is sparse data on novel modalities. Sometimes what happens, when the experiment is done and sparse data, that means some values are missing or majority of the values are zero. So such type of data can be generated during the study. So this is again another challenge, and basically this is applicable for mostly biologics and peptides. Such type of formulations, it is applicable. Then complex degradation kinetics. Some drugs have a very complex pathway of degradation, and not only one way, but multiple ways of degradation can exist. So it could be very difficult to address the degradation pathway for that one particular formulation or a drug substance. Then secondly, of course, it is their chromatography. Now this challenge is not considered as a challenge because there are n number of sophisticated instruments nowadays available with us. But yes, these are the points which can be considered in the challenges to the stability studies. So now coming to slowly to the AI, how AI integrates into the stability testing. Now we all know that artificial intelligence is everywhere now, and artificial intelligence is all about the predictions. So of course, the stability we can also predict for the drug substances as drug products. So what I mean, how integrates into the stability study? The first of all, data centralization. The AI, with the help of AI, we can have the chemical structures, the results which are obtained from stress as well as force degradation studies, and the images, for example, chromatogram images, and the packaging information. These all information can be at one platform, so that various different AI tools can be used and the predictions can be done. And the predictive modeling, for we all know that machine learning is the best tool for predictive modeling. So basically, machine learning is requires a large number of data. So that source of data has to be verified first. Third one is vision automation. There are few AI cameras which can monitor the physical changes of the drug substances as well as drug products. And the fourth one is the regulatory automation. Now, there is one technique, I mean, the tool, AI tool, that is NLP, natural language processing. So, this NLP system can be used to draft ICH compliant reports because NLP is it can perform a text analysis as well as translation. Then it is also integrate with the voice also. So it is very wonderful technique, so that it can have a sentiment analysis also of, so human and machine can be interfaced between interfaced with the NLP system, so that report writing can also be easily and it will be faster. The outcome is basically, it is the testing, stability testing is maybe very faster, cheaper and more accurate, while ensuring the safety and compliance of the data. The AI can be integrates into the stability testing. Now, these are core methods available in AI. Just to name these: machine learning, sometimes linear logistic regression, random forest. These are regression methods. Random forest, gradient boosting. These can be used for developing a AI model and for prediction also. Then some deep learning techniques. Deep learning techniques like feed forward nets. The feed forward net is widely applied across various fields for involving pattern recognition. So pattern recognition as well as classification is this particular feed forward nets is useful. So basically, some drugs have a specific pattern of degradation. So that can be, I mean, for identification, for prediction of that degradation pattern, this feed forward nets can be useful. Then RNN is also a good tool for for prediction of the stability studies, because it is applicable to the sequential data, and it can have a shorter forecast. So data is generated. We can have a help. I mean, we can get the data from the earlier studies also, and then we can, based on that sequential data, we can predict for shorter forecast. So what could be the probable expiration period and all. So this can reduce the time for prediction. Then there are different hybrid models available. The active learning and experimental design. Yes, active learning is basically, it is involved in design of experiment, and it efficiently uses all parameters for design. You can have a strategic design of uh, protocol for designing the stability testing. So strategic design of protocol can be possible by these active learning and experimental designs. So these were some tools. Now data sources and types. From where we should, what sort of data is required for giving to the AI model building. So stability time series, assay potency, impurity, accelerated state studies, high temperature, humidity samples, all quality attributes data parameters can be considered for developing a AI model for prediction of the stability, right? Again, secondly, the HPLC chromatograms, LCMS, in FTIR spectras, then manufacturing process parameters, all batch records, and some formulation related data like excipient, pH, container closure system. These all data can be considered in depth for developing, training our model. Now, these are all automated analytical interpretations can be possible. Chromatogram peak detection. So, by different techniques, we can have the peak detection of the chromatogram. Then spectral deconvolutions, chemometric methods can be applicable, and some pattern of degradation can also be studied. Then image analysis can also be possible for physical stability. So crystallization, the state of crystallization can also be predicted by using these AI tools. So this is how AI may contribute for the stability testing. But of course, it has to be validated. Whatever model is generated by using AI tools for prediction of stability, it has to be it has to have complete validation, and some metrics like RMSE, that means root mean square error, then MAE, that is minimum MAE is mean absolute error, and then ROC, AUC, all these terms can be used for validating the AI model. Then external validation on independent batches as well as prospective verification studies is important for validating the models. Stress test models across formulation variety as well as manufacturing sites. So basically, the AI models, if the AI tools are using, then we have to have validation of that particular thing by using these matrices. So this is how AI can contribute into the stability prediction. Now, some best practices are to be considered. First of all, we have to see that the data which we are taking is accurate or not, it is good quality data or not. Then it should have representativeness across batches as well as storage conditions. So for data is to be divided into two sections, as we all know, that training and test sets. So that has to be ensured that the quality as well as representative. The training set data should represent in the test set data, and test set data should be represent the training set data. So then only a meaningful good AI model can be generated. And secondly, predictable models where decision affect patient safety. So patient safety is to be considered, and then only that particular data is considered for the building a model. Cross-functional teams, say for example, analytical scientist, data scientist, quality regulatory. So we can change the analytical scientist, as earlier we used to do it, and the data which is obtained from exchanging the analytical scientist or changing the data scientist quality as well as some different experiments, that data should be chosen for the prediction of the stability. Expiration period. So tools and libraries which can be used. These are some most popular tools, open source tool, scikit-learn, TensorFlow, PyTorch, XGBoost, Prophet. These are all tools we can use for regression analysis as well as prediction. Chemometric methods, partial least square, all these chemometric method, and machine learning is a good platform for developing a predictive model. So these are few tools which we can. There are n number of tools available. We can have any tool which is more robust and reliable, accurate. So after that, there are some risks available with this ethical consideration. Sometimes overfitting may observe. Overfitting may limit the stability data, and sometimes we can get false results also. So for that, we need to be really vigilant about the model building. Then hidden confounders. Sometimes during the, because the data considered for prediction in AI is huge. So what happens sometimes, there is one third factor which is contributing to give the wrong results. Suppose we have considered 360 parameters, but there is, other than 360 parameters, there is one parameter which is interfering in predicting our models. So such type of hidden confounders are available. I mean, they may arise during the model building, so that has to be taken care. That is the another risk or limitation for this AI in stability prediction. Then the third one is regulatory pushback if the model are opaque or insufficient validated. Right. So if it is poorly validated compound and it is going ahead, then regulatory bodies will going to not. I mean, they are not going to accept that particular thing for making the product successful. So data privacy, IP protection, these are all parameters of risk is there, because data is used from a variety of source, and original data is considered for developing these models. So these are all risk limitations, and sometimes it is taken into consideration for ethical considerations. Now, FDA perspective on AI ML model is FDA expects transparency. They want entire data when they demand any, I mean, when any pharma industry is submitting the data and which is predicted by the AI model, they want entire data along with the models, validation results, and statistical results. So this is one thing. AI acceptable for decision supports, trend analysis, and early risk detection. So, for our understanding, we can use AI. This is what the FDA perspectives. So, trend analysis. If this particular class of drug is degrading this way, or degrading by pathway A as well as pathway B, then this trend can be analyzed for the rest of the formulation. So, for analyzing this trend, we can get the help of FDA. Direct expiry assignment still requires conformity long-term stability data. So FDA do not rely on only predictive modeling. They require, of course, long-term stability data for the experimental validations, right? So this is all about the perspective of FDA. That means here we can conclude from that FDA is not solely depend on the AIML, and it has to be there like that only. So where AI is generally acceptable, as I told you, the trend analysis is can be done. Early prediction to prioritize formulation. We can predict what type of excipients can be used. We can prioritize the formulation, as well as we can prioritize the packaging systems also. In that case, generally AI is acceptable. Supporting justification for bracketing and strategy, because we are reducing the stability. Bracketing, we have understood that reduce stability testing. So for making a justification, we can get the help of AI models, predicted AI models. Then the last one is detecting anomalous batches, unexpected degradation behavior. If it is there, then predictive modeling you can use, and then you can come up with the results because of what it is unexpectedly degrading. So AI can be acceptable in these aspects. Next is where AI is not acceptable. Assigning initial commercial shelf life. Shelf life cannot be assigned only based on the AI. Experimental validation is must for that. Then, of course, the waiving required long-term stability studies. Of course, FDA demands the long-term stability studies. Then justifying OOS results. in that case, AI is not acceptable. Replacing validated analytical method, no, it is not acceptable because the developed method is already validated, is applicable to the samples. That method cannot be replaced easily. So in that case, AI is not acceptable here for the stability is concerned. Now, lastly, regulatory expectations for AI model validations. Regulatory expectations are training data relevant to the product process and storage. So data which is used for training the model, it has to have concern with the product as well as process, that means the process of manufacturing as well as the storage condition. So this type of data is required for the regulatory bodies if the AI model is used. Then independent validation using unseen batches as well as time points. So some batches you require independent validation, not in a group. So that is required for the regulatory expectation. Then define performance metrics, acceptance criteria, on what metrics you have developed this AI tool, AI model, that they need to know. Acceptance criteria, all statistical results and their acceptance criteria they want, and ongoing monitoring as well as revalidation. If in case it is asked for, then one has to submit all these requirement to the regulatory bodies. So this is a scenario in AI. Now document. What is to be document here? Data sources. I just told you that from where you have collected the data. Then pre-processing steps and exclusion. If there is any exclusion from the data, the justification is always required for why you have excluded this data from the model building. Then model type, rational for selection of a data, rational for making it available in training as well as test set. Then performance results, limitations, uncertainty handling, and all these documented things are required. So these are all different documents are important for regulatory submission. But this AI, FDA always says that AI is not the only acceptable for having the compliance from the regulatory body. Their experimental validations are again important for the compliance. Now, from this key regulatory takeaways, AI is acceptable as scientifically justified decision support tool. So it is a tool for decisions, making a decisions, not the final output. So acceptable for only making decisions. Expiry assignment must still rely on real-time stability data. This is a takeaway. So no expiry using AI tools can be finalized. Experimental stability data is always required. Clear boundaries between predictions and confirmations we have to decide. So if the model is applicable to one particular shelf life, their prediction limitations are to be considered or are to be given importance. Then early dialogue with the regulators is strongly recommended. Always, once the AI is used, you always have to be in communication with the regulatory affairs, regulatory experts. So these are basically takeaways from the today's AI involvement in the this one stability predictions. So this was all about from my side, and you can reach out to me on this email ID as well as phone number. And thank you very much.
Thank you, sir. Now the session is open for question, answer, and discussion. Sir, I have one question.
Yes.
What skill should pharmacy student develop to work at the intersection of regulatory affairs and AI?
Yes, of course. Basically, the student, as far as students are concerned, students should know all basic guidelines. Very first of all, ICH guidelines, all stability guidelines, their nomenclatures, because many times the students goes for interview having, for this such type of departments, uh, then students are more reluctant about the nomenclature of the guidelines. So it is very important to read all the guidelines thoroughly, and understood that guidelines very carefully, and then what all possible guidelines to be implemented at college level that can be implemented, then only it comes into the practice and automatically it could be spoken during the interview. So I feel that the guidelines are important, and that is most crucial part of all pharma industry. And the recruiters are also searching for people who are knowing this particular thing, so that they can have ready personals for their work.
Okay, sir. Thank you.
Yeah.
Yeah, dear delegates, any other question?
I don't have the question, but I can make certain comments that Dr. Vishal has elaborated many things which are really related to the theme of our conference, and he has explained very nicely all the aspects of and the impurities and AI, everything. Thank you, doctor.
Thank you, sir. Thank you so much.
Sir, one question received from
Hmm.
YouTube state.
Hmm.
How can AI-based stability prediction be aligned with current ICS regulatory expectations?
Uh, See, AI is applicable to all the fields, not only it is only for current established guidelines. See, AI tools are generalized. We can apply it to pharmacy also. Even we can apply it to the data science, market research, right? So these tools can be used. Only thing is that the data which we are using or which we are taking for building a model, that is important. So of course AI can use for as per AI can be aligned with the latest guidelines which are introduced, but the thing is that one has to be vigilant. Therefore now multidisciplinary approach is coming into the picture. Every pharma student has to know all basic tools available in the AI, and that is why I think the NEP next syllabus which is going to be implemented, the AI is implemented. I mean, AI is incorporated. So yes, definitely AI can be used as and that can be aligned with the latest
ICS guidelines.
guidelines which are introduced nowadays.
Yes, thank you, sir. Okay, thank you, sir. So,
Okay, thank you so much.
yeah, sir has highlighted the session with the evolving regulatory expectation governing the stability testing with emphasis on ICH guideline beginning from 1993, data integrity and lifestyle management of pharmaceutical product. Sir has also explored how artificial intelligence and advanced analytics are transforming stability studies by enabling predictive modeling, analytics, risk-based decision making, and reduction in time and cost while ensuring regulatory compliance. With the conclusion of concluding the session, Sir has given one very nice balanced approach, that is combining regulatory knowledge with emerging digital tools, which is essential for pharmaceutical professional to ensure product quality, patient safety, and global regulatory acceptance in an increasingly data-driven world. Thank you, sir. On behalf of the organizing committee and our institution, I sincerely thank our resource person for sharing valuable knowledge, experience, and time with us. The clarity of concept and practical perspective presented today here will surely benefit all of us in our academic and professional journey. Thank you, sir.
Thank you so much.
As per the sir, anything else, Aman sir?
No, the oral presentation will going to start from 2 p.m.
Okay, so as per today's schedule, second day of the SNJB Pharmacon 25, now there is time of lunch break. So the oral presentation, scientific presentation will start at 2 p.m. Thank you.
Okay. Thank you. Thank you, Vishal.
Thank you so much, sir. Thank you.
Thank you. Thank you.
तुम्ही जॉईन आहे का?
ठीक है डॉक्टर गाड़ी क्रॉस करो ये यहीं पर है इनकी।
किती वाढीये ये तो माइक है ना माइक है गिनतो पण करेक्ट प्रोग्राम चालू करायचं बटन पुढे हेच बटन डायरेक्ट वाचून जातो विवेकानंद सर
Yes, sir.
hello can you hear me
Yes, sir. Very much.
hello
Yes, sir. Very much. I can hear you.
hello
I can hear
Hello.
you.
Hello.
Yes, you are audible.
It's a first one. Vivekanand.
Yeah.
Hello.
Hello, sir, is it audible?
Yes, yes. Can you hear us?
Yeah, we
Yes,
can
yes,
hear you.
yes.
Yes. Good evening. हो. तिथं प्रयत्न करा होतं का येतोय आवाज सर
हो, आवाज येतो.
येते सर.
ओके
येस येतो.
सर दिसले
गुड आफ्टरनून कटकुटवार सर.
Good afternoon, Amrutkar sir, Ashok Katkurwar sir. We welcome you for this SNJB Pharmacon 25.
थँक्यू व्हेरी गुड आफ्टरनून टू वन अँड ऑल
Good afternoon, sir. Good afternoon.
Good afternoon.
Shall we start, sir?
येस
Yes, we will. It's already 2 o'clock. Please start.
Yeah. Respected dignitaries, honorable judges, esteemed faculty members, research scholars, presenters, and all dear participants, a very good afternoon to all of you. We now move on to the one of the key academic segment of SNJB Pharmacon 25, the oral presentation session, which provides a valuable platform for young researchers and students to present their scientific work and receive expert feedback from both industry and academia. It's my privilege to formally welcome our esteemed judges for the oral presentation session. We are honored to have with us Mr. Ashok Katkurwar sir from Glenmark Pharmaceuticals, who brings rich industrial experience and practical insight into pharmaceutical formulation development. We are also privileged to welcome Dr. Sunil Amrutkar sir, Principal, Dr. M.S. Gosavi College of Pharmaceutical Education and Research, Nashik, a distinguished academician and administrator with vast experience in pharmaceutical education and research. On behalf of SNJB Sriman Suresh Dada Jain College of Pharmacy, Management Principal, and the organizing committee of SNJB Pharmacon, I extend a warm and sincere welcome to both of our respected judges and thank them for sparing their valuable time to evaluate the presentations. I now request Dr. V.A. Chatpalliwar sir to kindly take over the proceedings of the oral presentation session and brief the participants about the presentation guidelines and further conduct of the session. Thank you all, and I wish all the presenters all the very best. Thank you.
Thank you, Dr. Manoj. We have come to the last part of this platform of SNJB Pharmacon for the research to present am I audible now?
Yes, sir. Now it's
Now it is audible. Okay, sir.
Okay. Yeah. Sorry for the misconvenience. I was disconnected for a small moment. I welcome you all for this last session of SNJB PharmaCon 2025. It has been our constant efforts to provide a good platform for the young researchers to present their findings through this. This year we are going on, hence we present all were based on some findings, some research findings. There were no review articles as it was declared earlier. I am thankful to you for all the participants who have submitted their abstracts, but we are the invitation to or provide a chance for only few candidates to present their findings. I'll read out their names and then the rules will be followed. So please be ready with your presentations. I will read out the sequence to you as it is sequenced, and the list is also available the judges. The first presenter here, able, she may keep her presentation ready, followed by second presenter, Ms. Hitika Deshmukh. Third will be Ms. Snehal Nagpure. Fourth will be Mr. Aditya Dongre. Seventh will be
There was some issue with voice, you know.
Sir, Chatpalwar sir, your voice is breaking, sir.
Sonali Patil. Should I repeat the list?
Yes, sir.
Should I repeat this? Hitika Deshmukh. List. I will take a moment to change my setting. There is regarding my
Hello. Some internet issue is going on.
Hello, sir. Hello.
Okay.
Hello. Please bear with us. Some internet issues is going on. We are in the process of correcting it.
Okay, sir.
hello. I am back. I hope now that my voice is audible.
Yes, sir.
Okay. Okay. So sorry for the inconvenience caused. I will repeat the list of selected candidates for their presentation. You may follow the sequence. One by one, the candidates may keep their presentations ready. First candidate is Miss Pratiksha Venkatpati. Second is Miss Hitika Deshmukh. Third candidate is Miss Snehal Nagpure. Fourth candidate is Mr. Aditya Dongre. Fifth candidate is Miss Nidhi Patel. Sixth candidate is Miss Namrata Desai. Seventh candidate is Miss Sonali Patil. Eighth candidate is Mr. Varun Joshi. Ninth candidate is Mr. Viresh Kumar. There are these 9 presentations which have been selected for presenting their findings. I may brief you with the rules of the presentation. Some guidelines were forwarded earlier. Every candidate will be given 10 minutes for their presentation, 5 minutes for defending their presentation. Judges will be asking the questions only for 5 minutes. We'll shift to another candidate. Results will be declared post 10 minutes last presentation. After completion of all the presentations, all 9 presentations, judges will take few moments and then they will declare their results. There are 3 prizes to be given, 3 ranks to be given. And I hope all the scientists and all the young presenters will take in their strides and accept whatever decisions are given. This is just a competition, a part of a journey. It's a learning phase. So everybody should be taking it in their strides. And I wish all the best for your presentations. May I have the first presentation, Miss Pratiksha Venkatpati? Is she available?
Yes, sir, I am available.
Okay.
Am I audible?
Yeah, you are audible. I may ask the technical team to allow you to share your slide. The most important information for the presentation is you are being given only 10 minutes. Our technical team is monitoring the presentations. The time will start from your first slide. The moment you start speaking, your clock will be ticking and there will be a bell given by our technical team. May I ask our technical team to give a sample of that bell? May I ask the technical team? Hello?
Is the sound coming?
Yeah, no, it was not audible.
Yes, sir.
No. Our technical team will be interrupting as soon as the 10 minutes are over and the judges will be overtaking to question. So,
that's fine.
Ms. Pratiksha, may I request you to start your presentation?
Yes. Is my slide visible? Hello.
Yes.
Is my slide visible?
Yes.
Yes, Pratiksha.
Hello.
Good morning, everyone. Myself, I am Pratisha from Indus Biotech, Department of Scientific Affairs, Pune. So,
Go for presentation mode. We are looking 2 slides. Go for presentation mode.
yes, 1 second. Hello. Is it okay now?
Yes.
Yes, it's okay.
Yeah. Myself, I am Pratisha Venkatpati from Department of Scientific Affairs, Indus Biotech. Today, I will be presenting the effects of fenugreek seed extract, that is mainly called glycosides. Its effects on FCA-induced arthritis. So let's get into the introduction of what is rheumatoid arthritis. Like this rheumatoid arthritis is generally an chronic autoimmune inflammatory diseases where the symptoms involve. Hello.
Yeah, you are audible.
Yeah, you are audible.
Yeah.
Continue.
I'm not able to see my screen. 1 second. Just give me a few minutes.
We are able to see the screen.
Now it is visible to us.
Hello.
Yes, it is visible.
Yeah. Sorry for the inconvenience. What is rheumatoid arthritis? It is like an autoimmune disorder where it is seen in mostly in women compared to men, and like in worldwide, it is like compared to India in Western countries, there are rheumatoid arthritis diseases, but due to their treatment and their facilities, diagnosis. Things they are able to detect it very early, but in India there are less what to tell it, like diagnosis methods are like not that accurate, and due to this like many people, and the cost of treatment is also like very much like India, and many peoples are not like affordable to the treatment. And there are some treatment like present treatment of options for this rheumatoid arthritis are like NSAIDs and corticosteroids, and like DMRs like methotrexate, leflunomide. And these are there are some for these synthetic drugs, there are some like drawbacks for these synthetic drugs. Like when people start consuming these drugs in the long term, they got like toxicity. It mainly affects liver, kidney. Why it affects liver? Because it as we have, when we take it in oral form, there is a there obviously there will be a first pass metabolism. Due to that, the liver may get affected, and there is this rheumatoid arthritis mainly it is involved in it involves in immune system. So there is like when we take synthetic drugs to suppress this rheumatoid arthritis, there is this chance of like immunodeficiency. People will there is a chance of people getting infected to many diseases or viral infections easily, because their immune system will depress that immune system. And next thing is like it is like very costly, and that next thing is like they can't provide complete cure for that disease. And next we'll see with why there is a need of animal model, because we in this era we have like different technologies, speaking in a regulatory point of view. Before getting into human clinical trials, we need like particular safety data to get into the human clinical trials. And other seeing in a disease point of view, creating or getting rheumatoid arteries suffering humans, it is very difficult. Either we see the patients who have like their disease is like it is highly prolongated, or progression of the diseases will be there. So starting like getting early stage of disease is very difficult. And next is like when by the time when we discover the people is suffering from like rheumatoid arthritis, so the disease will be in advanced stage. And next is like the early rheumatoid arthritis disease stage, tissue samples are not available, so we can't do any study on this tissue. And next is like Why this animal study is selected? Because this animal study and this immune system involvement in this animal model for this rheumatoid arthritis, it's similar to like what happens in human body. And the other option which we have to like create rheumatoid arthritis is like surgical methods are available for that, but it becomes like chronic. So this is why we just follow animal model. And the first objective of this study is like to uh, evaluate the anti-arthritic activity of phenoglyco extract on FCA induced arthritis. This FCA induced arthritis is a like one of the model to the to study the anti-arthritic activity. And next is next secondary objective is to study like how this subchronal bone is important in anti-arthritic activity, and to examine the complex interactions with this arthritic progression. And next is before initiating any study, we just need a plan of work and plan of action and what are the methods to be followed. And before starting this experiment, we have to select the plant material. The plant material which we have selected is fenugreek seed. So this seed we have to get authenticated. Then we have isolated this phytoconstituent by HPLC. And next, after once it is done, we have to get ethical permission from the place where we are doing with the preclinical study and their particular ethical committee. And first thing we have to do with the safety. Safety in the sense acute toxicity testing should be done. And next, we'll go with what is the method for induced arthritis and how we are going to treat that. First thing is like as every study is done, animals should be like before starting of the study, before 1 week, animals should be acclimated to the laboratory conditions. And before the day of experiment, animals should be starved for 24 hours before the study. After that, this FCA is called as Friends Fion's complete adjuvant. We'll inject this 0.1 ml into the tail of the animal. Here we are selecting female Wister rat, because this female Wister rat develop this arthritis or any diseases easily. And if we select female rats, we can study the hormonal changes which may occur due to our drug, or like that hormonal changes we can also see. And next, once the like adjuvant is induced to the animal, next we have to monitor for the toxicity studies. And next we have to monitor whether the arthritis is developed or not. And after like 21st day of the inducing adjuvant, we have to give other shot of 0.1 ml of FCA to make it chronic. Why we do this booster dose is because once from the day of inducing FCA till 14th day, there is a chance of like decreasing the arthritis. So if we give the booster dose, so we can maintain it like till the end of the study. And after, like when we confirm that arthritis is developed by the 24th day, and next 25th day, we'll be treating it with phenoglyco products. And the standard we used here is leflunomide 10 mg per kg, and our product like phenoglyco, we made it into 3 doses, 3 mg, 10 mg, and 30 mg per kg. And next, after once at the end of the study, results were analyzed and parameters were measured on like weekly basis. So this is the method of FCA induced arthritis. And next we'll go with what were the parameters measured. Here we in vivo parameters were like body weight, food intake, paw volume were measured by different techniques like Vaughan, Harris, Randello. And main thing here in vivo parameters is like paw withdrawal latency and edema. And next is in vitro parameters. In vitro parameter, all blood parameters and organ weight, liver weight will be there, SGOT, SGPT. And then TNF alpha inhibitors, this plays a key role in like arthritis. And next, once it is done, We have done with the histopathology of tibosteral joint of the rat, because this why we specifically to do histopathology with this joint is like this fluid called as synovial. It is like more in on this joint, more fluid content is there on this joint, so we specifically take this joint for the histopathology. And here are some few results which we got. So if we see left hind paw volume and right hind paw volume, when it is compared with this arthritis control, fen glyco 30 mg and fen glyco 10 mg is decreasing the paw volume compared to the standard. And both in the same both right and left hand, it is like decreasing significantly. And next is like TNF alpha. As I said, TNF alpha plays main key role in this arthritis, people suffering or induced arthritis. Here in arthritis control, there is like more increase of TNF alpha, but when it is compared with standard fen glyco 30 mg and fen glyco 10 mg, it's significantly decreasing the levels of TNF alpha. And change in body weight, generally when there is a disease control group, once the animals get affected by the disease, there we can see animal is having like decreased body weight, not consuming food properly, and these parameters will be there. And when you compare fen glyco 30 mg and fen glyco 10 mg with the standard, there is
10
like
minutes over.
just 2 minutes are only, last slide is there. And next, and next is joint diameter. This joint diameter, when it is seen in like disease
Yeah, I
control.
may request Miss to stop her presentation, and whatever she has presented would be judged and questioned by the respected judges. Over to the judges, please.
Yeah. Ms. Pratiksha,
Yes, sir.
you use the fenugreek extract for arthritis testing, no?
Yes, sir.
What is the chemical constituent of this fenugreek, which is responsible for as anti-arthritis?
It is a glycoside based.
Glycoside base. There is no specific chemical constituent.
It is like glycoside, which we have like 80% of these glycosides. Based on that, we have done this study.
The question is very easy, simple, because it is already reported what are the chemical constituents of fenugreek.
Huh.
So I'm just asking which are the chemical constituents present in
Yeah.
fenugreek. Among that, which is responsible as an anti-arthritis?
In this phenylphalates, we have alkaloids, saponins, and while doing this HPLC, we have removed these saponins and alkaloids. Alkaloids is response.
Okay. You have mentioned you have used the female rat only, and
Yes, sir.
which kind of hormonal changes do take place? Can you explain?
Sir, the hormonal changes in the sense like doing this.
Which
For
hormonal
example, we.
take place? There are so many hormones in a body which are specifically get changes when you are tested for this arthritis that you have mentioned. We have used only female rat because there are hormonal changes. So which kind of hormonal changes?
Like it's not like hormonal changes. Generally, why we have taken female rats is like prevalence in humans is also like compared to men, women is having a chances of getting more arthritis. So based on that, we have taken. Another point is like if there is any like how to tell, if there is any changes.
Hi, sir.
So, Pratiksha, tell me which extract category is alcohol, ethanol, or water? What you are using as far as the extraction is concerned for these fenugreek seeds?
Sorry,
How you have
sir.
done the extraction?
Hello.
How you report the extract? Hello.
Hello, am I audible, sir?
Yes, yes. How you remove the extract? Which extract you used?
We have used ethanolic extract.
Okay.
Ethanolic extract by ethanolic using ethanol.
And now, when you compare with the NSAID of steroid, where we stand as far as the clinical efficacy is concerned of this fenugreek seed vis-a-vis NSAID and steroids?
Sorry, sir, can you repeat the question again?
As per the arthritis effect is concerned, where we stand fenugreek extract vis-a-vis against NSAID and your steroids,
Sir, actually, NSAIDs, corticosteroids are there are some adverse events reported on these NSAIDs and corticosteroids as they cause gastric ulcer. In our study, we did we have isolated the stomach and seen for this ulcer formation and everything, but we didn't see any ulcer formation in that. So we can, based on this, we can tell that our product is not getting any adverse events or something like that, causing gastric ulcers.
So what will be your dosage form in the days to come when you formulate this? Now, what will be your dosage form for this, and what will be the strength in tentative?
So once the clinical here in when we see in the animal study, mainly 10 mg and 30 mg is like providing significant effect in controlling arthritis. So when it comes to human studies, we may change. We may take the dose based on this.
Okay. Slide number 7, you have mentioned right hind and left hind. What is that hind stand for?
Sorry, sir?
On 7 number slide, you have mentioned right hind, HIND, and left HIND. What is this?
It is actually like measuring the paw volume, sir. Like whenever there is arthritis, there will be increasing in the paw volume. Like because of this immune reactions and T cell activation, interleukin activation, there will be the swelling. That hind is the thing is like rats. How to tell paw?
May I interrupt you people?
Yeah.
We are already exhausted the time allotted for the candidate. Yeah, thank you, Ms. Pratiksha Venkatpati. Your presentation was on fenugreek extracts. It is quite encouraging. The results were quite encouraging. Okay, we may wish you all the best.
Thank you.
We will shift our attention to next candidate, Miss Hitika Deshmukh. Is she available?
Yes, sir.
Yeah. Ms. Hitika Deshmukh,
Yes,
if you
sir,
are ready with
available.
your presentation, you are requested to share your slides. You will be
Yes.
given 10 minutes for your presentation, and further 5 minutes will be given to the judges to cross you.
Okay, sir.
Yeah, we have to see how do you talk. In last case, we didn't observe any video. Candidate, we didn't see the candidate last.
Yeah. Will you please put yourself on the video?
And in last presentation, we didn't see any conclusion. Last slide is very important. What is the way forward? What is the conclusion?
Because of the time constraints, I had to interrupt.
Okay, go ahead. Where is the candidate number 2nd?
Candidate number 2nd, Ms. Hitika Deshmukh, share your slides.
Yes, sir.
Yes.
Sir, is the slide available?
Yeah, but it is, you're sharing your mobile. It won't be read comfortably by the judges.
Your presentation.
Put it into your presentation mode.
Is it a PPT?
Yes, sir, PPT.
Put it in the presentation mode.
Yeah. Okay, good.
Yes. Yes. Your time starts now.
Yes.
Yes, yes.
So, good afternoon, everyone. Myself, Hitika. My topic, stability indicating methods. So firstly, why stability matters in assisted analytical method
You
for
are
the
not
AI
visible.
application, which
You
enter
are
the
not
outcomes of
visible.
the analysis.
Geetika, you are not visible.
Put yourself on the camera.
Madam bolo.
Yeah, it's okay now. Yeah, you may
Yes.
continue.
Yeah.
Yes, so stability forms the bedrock of the AI applications. In stability, this seems so. Firstly, it ensures the data. So firstly, it ensures the data integrity, in which the SIMs are in addition. Then third is the theoretical pillar, and with the conventional SIMs predominantly rely on the techniques like HPLC, LCMS, etc. Landscape shaping the SIM development, and that first the ICH Q1 guideline, in which the upcoming ICH Q1 guideline aims to globally harmonize the stability testing, and which is cutting edge of manufacturing and the analytical method and the development and quality control of drug. Third is the current guideline. The current integrating advanced AI technology to SIMS development. Then the challenges in the conventional SIEM development. First is the complex separations, in which the separation of APIs and their respective degradation products, which is the highly specified and often time-consuming methodologies. Second is the time-intensive studies. Studies in which the force degradation experiments and environmental. Then the AI game in SIEM development, which drives the speed up method development by predicting the most efficient chromatographic conditions. Then the degradation pattern analysis, in which the machine learning models, which analyze the degradation patterns and the force degradation studies. Then the real-time stability, in which the AI integrated with advanced sensors and data analyst, which helps in continuing the real-time. Then the integrating green chemistry and AI for sustainable SIMs. First is the eco-friendly method design, in which the AI is instrumental in designing the analytical method, which prioritize the environmental sustainability. Second is the hybrid techniques, in which the integration of AI with advanced spectroscopic and hyphenated methods result in the hybrid techniques to improve the efficiency and quality of the data. Then the global sustainability and the green chemistry, which aligns with the global movement towards more sustainable and the responsible for the pharmaceutical manufacturing practices. Then the future outlook, automation and hybrid AI approaches, in which the fully automated workflows, it helps to integrate the sample preparation, analysis, and comprehensive data interpretation. Second is the hybrid AI for biologics, which helps in combining the specific regulatory guidance, which regulatory framework and guidance specially tailored the transformation, in which first is the traditional SIEM workflow, which includes the manual process, time consuming, high cost, prone to human error and limited data analysis. And the second is AI assisted SIEM versus efficient, and fourth is the enhanced accuracy. Conclusion: embracing AI for the next generation stability assurance. First is accelerating pharmaceutical more robust, yeah, pharma industry is uniquely positioned to gain the specific competitive advantage in embracing the AI-driven approaches. Or is call to action, which
I may
invest
like
in
to
the
interrupt
AI
here. I may like to interrupt here.
capabilities and
Excuse me. Before the judges start cross-questioning, I would like to ask to the candidate, Ms. Hitika, is it
yes, sir.
a research article or abstract of a review? Yes,
It's an abstract of review.
it's
Hello.
a review. But the abstract sent to us didn't mention that it is a review. It was read and it was found that it is a research article. It appeared to be a research article. From that, it was the chance was given to you for presentation. I am sorry, Ms. Itika. This is if this is a review, it can't be considered for the competition. I am very much sorry for you. Although the abstract was reading that it seemed that it is a research article, but I am sorry.
Sir, actually 2 to 3 research articles were there from this topic, and I
No,
had
sorry, the
shortlisted.
abstract which you have sent was based on some research article. That's why it was selected. I'm sorry, no harms. You may try it next time. I may request the judges, I may appeal that this candidate and her presentation won't be considered for the competition. Thank you.
Thank you. Thank you, Chatwal.
Thank you, Ms. Sorry for today's chance. We may shift our attention towards the next candidate, Miss Nehal Nagpure. Miss Nehal Nagpure. Is she available? Miss Nehal Nagpure. Is she available?
She is not present
She is not present. Okay.
in the list of
We may,
participants.
yeah, we may shift our attention to Mr. Aditya Dongre. Is Mr. Aditya Dongre available? Hello, Mr. Aditya Dongre.
He is also not there in the participant list.
Just wait. Last call for Mr. Aditya Dongre. Not available. Okay, we call upon next candidate, Miss Nidhi Patel. Candidate number 5th, Miss Nidhi Patel.
She is also not there, sir, in the participant list.
Wait for a few minutes.
Namrata Desai is there.
Yeah, Miss Namrata Desai, are you there?
Yes, she is there.
Yes, sir, I'm there.
Yeah. Are you ready with your presentation?
Yes, sir.
Yeah, we may take up with your presentation. The candidates who have missed, Miss Nehal Nagpure, Mr. Aditya Dongre, and Miss Nidhi Patel will be given a chance if they are available, if time permits to us. Okay, so may I appeal our judges, our respected judges, to mark Miss Namrata Desai as the next presenter.
Yes.
Candidate number 6, Miss Namrata Desai. You may share your slides and all the best for your presentation.
Thank you, sir.
Okay.
So is my screen is visible?
Yes,
Yeah,
sir.
very much. Put yourself on camera. As per the requirement of the judges, you must put yourself on the camera.
Um,
Start your presentation.
Thank you, sir.
Yeah, put your camera on.
So I have on my camera.
Okay.
Switch on my camera. Am I visible? Hello.
Yeah.
Am I visible,
This-
sir?
You will get visible as you speak. Okay, you may start with your presentation.
So good afternoon, respected chairperson, esteemed panel members, and distinguished delegates. So I am Namrata Desai, and today I am presenting our research work entitled Clinical Evaluation of Glycemic Index of Defatted Fenugreek Seed Flakes, known as Fenuflex, in a healthy adult. So this study was conducted in collaboration with the Kalpana Bhaskar and her team at the Glycemic Index Research Unit, Temasek Polytechnic, Singapore. So these are my outlines of my presentation. So I have organized my presentation into 5 sections: backgrounds, which tells what is known and why the topic matters, followed by the rationale, so why this study was needed, then study design and methodology, which tells how we designed the study and how we conducted the study, followed by the results, so what we found, and the conclusion and the future directions. So let's begin with the background. So Indian dietary patterns typically consist of 70 to 80% carbohydrates, which serves as the primary energy source in a Indian diet. So the prominent carbohydrate source in these diets are often with the high glycemic index, which, for example, the white rice. So the dietary carbohydrates are broadly classified as the available carbohydrates, which are also known as the glycemic carbohydrates, which are metabolized in the body and which responsible for raise in the blood glucose upon consumption. Whereas the second classification of the dietary carbohydrates is the unavailable carbohydrates, which are not digested in the body and which may be fermented in the colon. So what is the glycemic index? So glycemic property or carbohydrate quality of the food assessed by the glycemic index, which is a relative ranking system that classifies the carbohydrate containing foods based on their ability to raise blood glucose level compared with the reference food. A reference food usually used as glucose. So this is known as the glycemic index. So how glycemic index is measured? So the International Organization for Standardization ranks food from scale 1 to 100 and use pure glucose as a reference standard. So GI is ranked on a scale of 0 to 100 with higher value indicating the faster, larger increase in the blood glucose. So to measure the GI of a food, human subjects are supposed to have a portion of the taste food containing the equivalent amount of carbohydrates as equivalent to the reference food. So usually reference food, pure glucose or a white bread used as a reference food. And afterwards, the blood samples from these participants were taken at in regular intervals over 2 hours to monitor changes in the blood sugar levels. So the area under the curve for each individual's blood glucose response is then calculated and compared to the reference food. So these are the classification given by the international standard of organization. So the based on the GI value, so foods are classified as the low, medium, and then high. So the GI value less than 55 is considered as a low. And these are the examples like barley, corn, whole wheats having the low GI. Whereas the GI 55 to 69 falls under the medium GI. So brown rice, muesli, noodles, soft drinks, honey falls under the medium GI foods. And the GI value greater than 70 falls under the high GI foods. For example, the white rice, bread, cookies. So foods with the low GI releases the carbohydrate slowly and helping to maintain the steady blood sugars and prolong the satiety, while high GI foods absorb more quickly, leading to sharper rise in the blood glucose. So understanding the GI can therefore guide healthier food choices, which supports for dietary planning and plays a crucial role in medical nutrition therapy, particularly in individuals managing the diabetes or seeking improved metabolic health. So diabetes is a the diabetes prevalence is alarming high among the Asian Indians, with the 7.3% of adults affected and 10.3% classified as a pre-diabetic. So partly due to an faulty dietary patterns. So food with the lower GI are recommended for the Asian Indians. So noodles and cookies are widely consumed bakery products enjoyed by a lot of people by all ages worldwide. Generally have a generally noodles and cookies are having a high glycemic index, approximately 70, so making them less suitable for diabetic and individuals managing the excess weight or obesity. So incorporation of the fenugreek seed powders into the food such as dhokla, upma, laddus has demonstrated significant reductions in the GI in the past literature. So, but they are having limitation like incorporation of fenugreek seed into a daily diet is having inherent bitterness and a poor palatability. So we have designed the defatted fenugreek seed flakes were developed as a palatable, which has 100% natural flakes and standardized from a form of a fenugreek seed, which are safe for human consumption as reported earlier. So these flakes are rich in fibers and proteins. So as name itself indicate the defatted. So we have removed the fat percent of the seed, and they are making them to rich in the fiber and protein, while containing the negligible carbohydrates and fats, allowing for the easy incorporation into the common recipes. So fenuflex therefore represents a novel functional ingredient with the potential to lower the glycemic index of high carbohydrate food preparations. So therefore, the present study aimed to determine the clinical evaluation of cookies and fenuflex cookies and noodles containing defatted fenugreek seed flakes for the glycemic index testing. So this is the study design and the methodology. So as per the international GI guidelines recommended minimum involvement of minimum 10 subjects, whereas in this study we have incorporated the 12 ULD volunteers to enhance the reliability and precision of the GI estimate. So 12 healthy volunteers with age between 19 to 60 years with BMI 18.5 to 25 kilograms per meter square were recruited from the Temasek Polytechnic, Singapore, while ensuring the ethics committee approval was taken, and while compliance with the Singapore Accreditation Council functional food testing laboratory. So so these 12 healthy volunteers were asked to for overnight fast for 10 to 12 hours. The post fasting coming to the centers, the food preparations like taste food, that is fenugreek cookies consist of 65.1 gram, and the fenugreek noodles 14.39 gram, and whereas the reference food 25 gram of dextrose, that is anhydrous powder in a 250 ml water. So they have to consume either taste food and the reference food on a different occasions, 2 or 3 occasions within a 15 minutes. Then post consumption, their fasting blood glucose were measured at 15-13 capillary when fasting blood glucose were measured at 15, 30, 45, 60, 90 and 120 minutes. And after this incremental area under the curve were measured, and the glycemic index of the food were calculated based on the standard formula, that is area under curve of taste food divided by the reference food multiplied by 100.
Last minute.
Okay, so the average 2 hours blood glucose response curves for the equal carbohydrate portions of the reference food, glucose and the taste food, that is fenugreek cookies and noodles, were shown here. So the reference food that is 100% glucose shown in the red was digested and produced a large rise in the blood glucose during the first 30 minutes and a large overall glycemic response as compared to the fenugreek cookies and noodles respectively. So the fenugreek cookies and noodles have a much lower peak blood glucose level and the low total glycemic response than compared to the reference food. So mean GI was found to be 47% in the fenugreek cookies and 54% in fenugreek noodles. So both these food preparation as classified as a low GI food. So it comes to the conclusion. So fenugreek shows the GI lowering effect. Both the food preparation shows the GI lowering effect, and which are attributed to the higher content of water soluble dietary fibers such as galactomannan, indicating their potential suitability for glycemic management. So fenugreek rich food exhibits low carbohydrate digestions, leading to improved postprandial glycemic control. And the soluble fiber in fenugreeks may reduce the risk of metabolic disorders. So low GI fenugreek food provides the energy, sustain energy, and reduce the post-meal fatigue. So dietary fiber content contributes to improve digestive and gut health. So in future, to glycemic response of these preparations can be checked in the pre-diabetic and diabetic population, and long-term intervention studies are needed to assess the glycemic control and insulin sensitivity, and this can be compared with the cookies and commercial cookies and bakery products. So I would like to thanks entire SNGB family to give us a chance to present our research work during the conference in this conference. And also we would like to acknowledge the glycemic index research unit Temasek Polytechnic Singapore for collaborating this work. Thank you.
Thank you, Ms. Namrata. Over to you.
Question answer.
Judges, respected judges.
Pallivar sir,
Yes.
should we ask question?
Yeah.
Yeah. Open slide number 9. Namrata,
Yes, sir.
where is the presentation? Slide number 9. You have mentioned some conclusion in that. How does fenugreek maintain stable blood glucose level? Just explain.
Sir, fenugreek actually contains the soluble fibers, basically such as galactomannan. So galactomannan forms the we can
Okay.
say,
Second question, of diabetes. How does it reduce of diabetes?
Sorry, sir.
You have mentioned second conclusion, reduce risk of diabetes. How does fenugreek reduces risk of diabetes? Explain.
Sir, it contains a high soluble fibers such as the galactomannan. So these forms the gels in the gut, so delaying the breakdown and absorption of carbohydrates. So absorption and breakdown, which delays.
Ms. Namrata,
Huh?
the fibers will be only acting in the gut. But
Yes.
diabetes is a general problem, is a systemic problem. Type 2 diabetes is generally exerted due to glucose excretion from liver. So how does your statement that reduces risk of diabetes stand true?
Again, it is a disorder of carbohydrate metabolism. So it delays the absorption of carbohydrates, so which indirectly prevents the rapid blood sugar increase after meals.
So the cells will be starved of glucose and liver will be exerting a lot of glucose in turn in the bloodstream. The net effect will be increase in the glucose blood levels. Okay.
Okay, sir.
Over to judges, sir.
So, Namrata, what
Hmm.
you are using fenugreek flakes, right?
Yeah, sir.
Tell me, what is that chemical constant that helps in the reducing the sugar level?
So fibers, soluble fibers, sir. Galactomannan.
Okay, if that is the thing, what is the future? What dosage form you want to make it from the future? Because if you want to make some dosage form from these flakes, right? So, what is the preferred choice as per the dosage form from the future perspective is concerned?
So dosage form we can make it in a drink, or we can use as like it is a flex, no sir. As you can see here, it's a flex. So you can sprinkle it over a food preparations like poha and all, or you can use during the preparations, oats and.
So because having the bitterness, you can't have a much, you know. So whether from the pharmaceutical perspective, which dosage form you prefer, because you cannot have a much quantity as due to bitterness in the in the daily usage, right? So
Yeah, sir.
we wanted to have some dosage from these flakes, right? So how can
Yeah.
you, any thought on this?
I have no idea, sir. Sorry, but we can incorporate into the sport drinks or something, which can be.
Okay, madam. Over to Kitpaljar, sir. From our side, it is over.
No, no.
Good afternoon, sir.
Sir, Vivekanand sir, should we go for next candidate?
Yes. Sir.
Okay.
Next candidate is Miss Sonali Patil. Is she there?
Yeah. Start, madam. Sonali Patil, Snehal Patil. Please, Sonali Patil. Snehal.
Just
Due to some
confirm her
technical.
name.
Sonali Patil. Is she there? Miss Sonali Patil. If they are available, given a chance later. Next candidate is Mr. Varun Joshi. Is he there? Mr. Varun Joshi.
Yes, sir.
Yeah. Varun, you can share your screen.
Yes, sir. Just
In the meantime, I may appeal other candidates, those who have joined late, those who have missed their call, they may keep their presentations ready. After the sequence ends, we may give you a chance.
is it visible, sir, my screen?
Yeah, it's visible. You can
Yes.
start now.
Yes, sir. Good afternoon to all. Myself, Mr. Varun Joshi, from PhD research scholar from Department of Pharmacology, SNJB Sriman Surezada Jain College of Pharmacy, Chanwad. Today, I will be deliver an oral presentation on acute oral toxicity evaluation of the bioactive phytoconstituents in the female Spargew Dowley rats by following the OECD guideline 423 at SNJB Pharmacon 2025. So these are my contents of my study. So this is the rationale behind this study. So as the medicinal plants having the bioactive constituents, and it will be used in the various traditional methods in the healthcare system, and due to their healthcare therapeutic potentials. So the acute toxicity studies having the various crucial role for the identifying the possible toxic effects, estimating the safe dose ranges, and classify the substances. It will be based on their toxicity profile. So for this, we use our OECD guideline 423. So for which is the globally accepted protocol for evaluating the acute oral toxicity study. So in this OECD 423, there is the minimal number of animals it will be required, and these guidelines it will be used the ensuring the reliable safety data. So generally, female SD rats are preferred due to their higher sensitivity to toxic substances and consistent pathological responses. So in this present study, I was used the selective bioactive phytochemicals, that is phytochemical A and phytochemical B, and assessing their acute oral toxicity for to establishment of the toxicity at the higher dose levels. So these are my the objectives. Then, so acute oral toxicity testing. So the before starting of an acute oral toxicity testing, the animals were quarantined and acclimatized as per the CCSA norms before the before use in the experimental study. This is my protocol approval number. Then we are done the some standardization experimental procedures like pilot study, dose regimen we decide, then route of administration. Preferably we use the oral gavage route for the administration. Then the both the phytoconstituents we are solubilized in the 1% carboxymethyl cellulose solutions by using and distill water. Okay, and we prepare an suspension, and through the oral gavage route we administer these two phytoconstituent to the female rats. Then after that, from the standard operating procedures, we do the animal handling. After that, drug administration it will be takes place, and from that sample it will be collected and data recording were developed. So animal receives the NutriVet, rat mice feed pellets, and clean drinking water during the overall experiments. So animal housing and caging. So we use female SD rats of age between the 8 to 12 weeks, of weight around 180 to 220 gram, which are nulliporous and non-pregnant female rats we are used. Then 1 week acclimatization process we are given to these particular female rats. Then rats having the free access of standard pellet diet and drinking water. Then the bedding of that particular animals, it will be changed 2 times a week. Then temperature is around 22 to plus or minus 3 degrees Celsius it will be maintained, and relative humidity 30 to 70% it will be maintained in the animal house, and there is a 12 hours light and dark cycle it will be uh, monitored. So in the total study, there are the 15 animals we are involved in the 5, and the animals are differentiated into the 5 experimental groups. The animals are assigned as normal control group, then group A, who receives the 300 milligram per kg of phytoconstituent A substance, then group B, who receives the 2000 milligram per kg, as it will be also considered as an limit dose. Then group C, phytoconstituent B of 300 milligram per kg it will be received. Then and group D, the phytoconstituent B 2000 milligram per kg as a second limit dose it will be given. So administration is the single oral administration on the day 1 it will be given, and the each group containing the 3 animals it will be used. Total number of animals are 15. So this is the assessment study in which we are perform the general parameters like body weight, then cage size of side observations like changes in the skin and fur, eyes and mucous membrane changes, respiratory, circulatory, autonomic and central nervous system changes. Then after the day 15th, we selected the limit dose group animal and give the 65 milligram per kg deep anesthesia of thiopentone sodium on the day 15. Then the organs are dissected out and using 4% paraformaldehyde solutions, the animals are kept in that for to avoiding the autopsy and particularly the destruction of these cells. Then using an hematoxylin and eosin solution, the histopathology slides in that HE stain it will be used, and it will be under the Nikon Eclipse trinocular microscope with advanced specification. So in the resultant discussion, so every individual animal, it will be taken for the study was observed for the 14 days, and there is no any adverse effect were observed during the first 30 minutes, and periodically what during the first 24 hours for the group with a limit dose of 2000 milligram per kg. So in the body weight, the effect of phytoconstituent A and phytoconstituent B on the ST rat was observed from the day 0 to day 14, and the data were expressed as mean and standard error mean. The data were analyzed by using the two-way ANOVA test, by using the post hoc the net multiple comparison test, and in this body weight observation, we see that it will be shows the not significant results. So we can conclude that there is no any systemic toxicity will be happen during the dose administration to the animal. During the case side observation, there is no significant abnormalities in the test animals was found throughout the study of the 14 days. So histological examination, gross pathological examination of the post euthanasia will be revealed that there is no abnormalities in the vital organs like stomach. We are using the stomach, heart, ileum, lung, kidney, spleen, pancreas, ovaries, uterus, liver, and brain. So the organ weights and microscopic morphology fell within the normal physiological ranges, and there is no any evidence of discolorations, lesions, and particularly hypertrophy. There is no hypertrophy, it will be observed. So this is the histological images examination of brain, tissues of brain, heart, ileum, kidney, liver, lung, spleen, stomach, uterus of the negative control group, and it was shows an normal architecture of the normal architecture of the tissue. So in the second phase, we use an limit dose. We use an animals from the limit dose group of 2000 milligram per kg. In that we are using the same this organ from the organs were isolated, and it will be perfused with the 4% paraformaldehyde solution, and it will be for the to avoiding the autopsy, and it was shows that there is no any structural changes were observed in the particular histopathology of phytoconstituent A group. In the second group, phytoconstituent B, it will be also shows there is no any destruction between the architectural structure of the tissues of various organs. So this is the macroscopic examinations of the tissues. In that there is no discoloration, it will be observed. Then there is no any hypertrophy, it will be seen in the isolated organs. So this is my conclusion. So these present study conclude that selected bioactive phytoconstituent A and B following the acute oral administration in the SD rat under the experimentations of OECD guideline 423. So single doses of 300 milligram per kg and 200 milligram per kg did not produce any mortality, abnormal signs, and behavioral alteration. There is no adverse effect present on the body weight. So the absence of these toxic manifestation at the limit dose 1000 milligram per kg, it will be indicate that a wide safety margins for the both compounds, and it will be suggested that it can be classified as a low toxic substances, and these findings provide the preliminary evidence for the supporting the short-term safety of phytoconstituent A and B and justify their further investigation in the subacute chronic toxicity and pharmacological efficacy studies. So that's from my all side.
Okay. Thank you, Mr. Varun.
Okay.
Now, may I request the respected judges to cross-question Mr. Varun?
Okay, Varun Doshi, you have a very nice
Yes, sir.
presentation, but just I would like to ask, what is the purpose of your study? What you have done exactly and what is the purpose?
Okay, sir, purpose is to find out the toxic concentration of this phytoconstituent,
Which
and also,
phytoconstituent?
so these are the confidentials are there because there is no work it will be published.
Phytoconstituents.
So what is
No,
this
no.
PA you mentioned,
Yes.
PCA, PCB, what is because what are these a toxic study, what PCA, PCB? What is that
So
PCA,
these are,
what is PCB? From where you got the extract?
no, these are the bioactive constituent. So these are the purchased
Mr. Varun,
from the,
your
yes,
question,
sir.
your answer is right. These are the bioactive constituent. What is the application of this bioactive constituent?
Yes.
Particularly, these are the strong antioxidants in the property, and my
Why did
further
you
work
test
is
for the oral toxicity?
so for detect and safe dose for my further studies, which are used in the diabetic complications. So that's why I this performance.
In diabetic.
No. For studied in the further complications of these bioactive compounds in the diabetic complication study.
One question is very simple. What is the use of these bioactive compounds? For what you are using? Don't disclose the name, but what is the application of these bioactive compounds? Whether they are anti-diabetic, anti-arthritic, cardiovascular.
Yes,
What is the use?
Why you are doing this tox study? What is the
Why did you do this?
What is the next step?
My next step is performing animal study on the diabetic complications in which neuropathy, nephropathy, and cardiomyopathy. I have to conduct the experiments.
So the PC and PCB are anti-diabetic.
So these are they having the both anti-diabetic property, also has the strong antioxidant effect. I assess their effect by using the DPPS scavenging assay. So and from these assay, it will be shows the positive effect.
Suppose from tox perspective is okay, but if it doesn't perform from the diabetes perspective, what is the then next step? Have you done any initial preliminary study from the diabetes perspective for this?
Sir, I performed the in vitro alpha glucosidase and alpha amylase test, and these tests it will be shows the significant results for the further study.
So first, what I feel first is the therapeutic effectiveness, then comes your tox. So what you have shown is the tox, then you are doing therapeutic effectiveness like that.
Simply are you testing toxicity first and then you are going to test for therapeutic effect? Is it like that?
Yes, sir. Yes.
If it doesn't work, then your whole work is of no use. No?
Yes, sir. Yes.
Yes. Your contents were good, but
Okay. Okay, Mr. Varun.
Okay, Mr. Varun, thank you for your presentation. Thank you for your efforts and respecting the timings. I may thank our judges and ask him for to permit me to shift to another candidate. Mr. Snehal Nagpure is in the waiting. Miss Snehal Nagpure text me that she is available for her presentation, but she will have to wait for Mr. Viresh Kumar. He is next in the list. Mr. Viresh Kumar, are you ready with your presentations?
Yes, sir.
Okay. Would you please display your contents? Make it in a presentation mode.
Okay, sir. I'm just presenting.
I may repeat the rules. The final decision of the judges would be acceptable to everybody. This is a learning phase for everybody, and we must respect the judges, their choices. You are being given only 10 minutes for your presentation. If you are on your last slide, we may give you 1 more minute to complete. And please put yourself on the camera.
Sir, am I visible?
Yes, very much.
Presentation mode. Go to the presentation mode.
Go for presentation mode.
Yes, sir. Sir, is it visible now?
Yes, yes.
Yes. Good afternoon, everyone. My name is Viresh Kumar, and today I am here to present my research work on the formulation and characterization of folate conjugated doxorubicin and curcumin co-loaded pegylated liposomes for the effective management of prostate cancer. These are the table of contents, which includes introduction, compatibility studies, method of preparation, particle size and distribution, drug release profile, and cell viability studies. First of all, prostate cancer is a malignant tumor that develops in the walnut-shaped prostate gland located just below the bladder in men. Its growth is often influenced by many sex hormones, powerful sex hormones such as testosterone, which are often converted to dihydrotestosterone, which cause the prostate cells to enlarge. Current therapies for the prostate cancers include surgery, radiation, chemotherapy. However, chemotherapy faces significant challenges such as non-site specific drug delivery, which often causes toxicity to the normal and healthy tissues, which reduces the therapeutic effectiveness, and multi-drug resistance. Our work aims to overcome these limitations and increase the therapeutic effectiveness so that the not the healthy cells could be targeted, but the drug should only be delivered to the target site, that is to the cancer site, to improve the efficacy. After that, next slide. These are the here is the picture of a prostate cancer, where the prostate gland enlarges in the size in case of the prostate cancer. After that, these are the compatibility studies which were done using FTIR to check whether both the drugs were compatible or not. Here we can see that in the curcumin and doxorubicin, and after the curcumin and doxorubicin combined when taken under the FTIR, the characteristics peaks were visible, which confirm that both the drugs were compatible and can be used in a single formulation. After that, the compatibility studies were done using DSC, and here the different thermograms of doxorubicin, curcumin, and their combination were very much justified that they both were compatible and the characteristic peaks were visible. That means that these can be used in combination with each other. After that, the method of preparation for the formulation was the thin film hydration for the liposomes. The liposomes were prepared by the thin film hydration method, in which the lipids were dissolved in the organic solvent, and the solvent was evaporated using the rotary evaporator to form the thin film. And after that, the sonication was done, which forms the liposomes in a uniform size. After which the medium was hydrated with the use of 5% dextrose solution, and the further conjugation was done with the help of folate DSP. With the help of this conjugation, we can target our formulation to the target site, that is to the prostate cancer cells, as it has been shown that folate receptors are highly expressed in the prostate cancer cells. So folic acid could potentially be used so that it could go into the target site and interact with the folate receptors, such that the formulation should reach only the target site, and with the help of receptor mediated endocytosis, it goes into the prostate cancer cells, and the release of drug could only be in the cancer cells. Here is the particle size and distribution. The particle size of the formulation was obtained to be 269 nanometer with the PDI of 0.291. That ensures that the PDI was less than 0.3, such that its size was very much uniform and with a uniform PDI, and the size was also determined with the help of TEM images. Here we can see that the surface of the liposomes was smooth, spherical in shape, and the particle size under TEM that was around 375 nanometers, so that it could effectively go to the target size and could be and endocytosis, with the help of endocytosis, it could go into the target cells. Then it was the drug release profile. Here we can see the drug release profile of various formulations. First of all, the curcumin, alone. After that, the curcumin liposomes and the curcumin doxorubicin liposomes. Here we can see that when the curcumin was tested alone, the drug was released instantly and a very short duration of time. But after giving it in the form of liposomes, the sustained release was obtained. We can see that there was a sustained release of the drug over a prolonged period of time due to the pegylation which we have done, as the pegylation helps in overcoming the immune system such that it is not cleared out by the reticuloendothelial system and could retain in the circulation for a prolonged period of time. As we and it is justified by the drug release profile, where we can see that the formulation was sustained in the plasma drug concentration for a prolonged period of time. Here were the cell viability studies. With the help of cell viability studies, we have determined the IC50 value of the formulation, that was 56.49 micromolar, and it has also and it was tested for both 24 hours and 48 hours. After 48 hours, the cell viability of the prostate cancers was very much less, which justifies that both the drugs were very much potential when used in combination, which justifies that both the drugs can be used and effectively reduces the cancer, and which helps in the treatment. In conclusion, we can say that the study successfully developed folate targeted pegylated liposomes for co-delivery the doxorubicin and curcumin.
Excuse me. May
Yes, sir.
I ask one of the participant to mute her computer? Okay, you may continue.
Yes.
Sorry for the glitch.
Yes, sir. The study successfully developed folate-targeted pegylated liposomes for co-delivering both doxorubicin and curcumin. The stable formulation showed sustained release and enhanced cytotoxicity against the prostate cancer cells compared to individual drug formulations, suggesting a promising strategy for targeted combination therapy. Here we can see that in the prostate cancer, in the tumor microenvironment, when the targeted formulation was given, it reaches the specific site of the prostate cancer on which the folate receptors were present, and the folate conjugation which was done on the formulation, it interacts with the folate receptors, and with the help of receptor mediated endocytosis, the formulation goes inside the prostate cancer cells only and not the healthy cells, such that it acts only on the cancer cells, which helps in the treatment of prostate cancer. That's all, sir. Thank you.
Okay, Mr. Virish Kumar. I am thankful for respecting the timings. May I ask the respected judges to have their questions?
Okay, Mr. Viresh, you have a very nice presentation. Just I have one question.
Yes,
You have mentioned some compatibility study by FTIR. Can you explain that? How does
Yes.
we come to know that compatibility? Which kind of compatibility, and how IR helped you?
Yes, sir. Sir, first of all, in case of FTIR, we checks the characteristic peaks of the drugs. At a particular wavelength, the drug shows a
Okay, that we
characteristic
know,
peak.
IR we know very well.
Yes, sir.
You have mentioned compatibility. What you have tested of compatibility? Which kind of compatibility?
Okay, sir. Sir, first of all, when as we are taking both the drugs when given in combination in a formulation, there could be a chance that both the drugs could interact with each other and form a new product, which could be harmful, which could be beneficial, or which could have a different effect. So it has been tested with the using FTIR.
Which kind of interaction do you expect, chemical or physical?
Yes, sir. Sir,
which kind of interaction you are expecting, physical or chemical?
Sir, it could be chemical.
It could be chemical.
Yes, sir.
Then how
Yes.
IR helped you?
Sir, IR will help us that are the characteristic peaks of the individual drugs of the curcumin and doxorubicin. Are the characteristic peaks visible in when we have taken both of these drugs in combination or not? So IR helped us determining that both the characteristic peaks which were of the individual drugs, these were visible in the combination. So it justifies that both the drugs were sustained and can be used in combination and do not react with each other.
Yeah. So when
As a audience, I may intervene here. Whatever explanation the candidate has given and whatever spectra he is displaying, I can say that it is not satisfactory concluded that whether there is an interaction, physical interaction or chemical interaction, it is not conclusive. It is not to be studied by IR spectroscopy, I suppose, because these spectra which he is showing, they are not satisfying the purpose. What do you?
Viresh, you done any stability study for this combination?
Sir, I haven't done the stability studies yet, but we are moving to it. Current now we have done the cell viability studies, cell line studies. We are also planning to do the animal studies, but till now we have done up to the cell line studies only. We are also planning to do the stability studies
So
in
what is the
future.
dosage form and what is the storage condition for this?
Yes, sir.
What is the dosage form and what is the stability storage for this?
So the storage condition which is best suitable is 4 degrees Celsius for the formulation of at which it is most stable as per the literature I have studied.
4 degree.
Yes, sir, 4 degrees Celsius.
How can we maintain that 4 degrees then throughout the time? During transportation, during storage?
So it is a lyophilized formation, right?
Yes, sir. Yes, sir.
Okay, sir is having some questions.
So any individual combinations for curcumin is available in the market?
Yes, sir. Can you please pardon?
So individual formulation is available for the curcumin. Individual formulation is available at present.
Sir, the individual formulations for curcumin are not specifically available for the prostate cancer. But if we take out, if we go to the literature, then there are some potential studies which justifies that curcumin has potential effect on cancer. It can be used in combination with other drugs to enhance the sensitivity of other drugs. That's why we are using curcumin in combination.
Curcumin has shown some anti-cancer effect anywhere?
Yes, sir. It has some anti-cancer effects. It also
is anti-cancer.
Anti-cancer.
So you are expecting synergistic effect here?
Yes, sir. Yes, sir. Curcumin basically helps in chemosensitization of the drug, which
Okay.
helps in increasing the therapeutic efficacy.
Good.
Okay, good.
Good. Thank you. Okay.
Over to Chatkulwar, sir.
Okay, sir. Thank you, Mr. Viresh.
Thank you,
We
sir.
will move our attention towards next candidate.
Thank you very much, sir.
Is Miss Nehal Nagpure available now? May I call upon Miss Nehal Nagpure?
Yes, sir. Yes.
Okay. If you
Good
are
afternoon,
available,
sir. Yes.
good afternoon.
Yes, sir. Yes. Good
Please
afternoon, sir.
put on your presentation. Please share your
Yes,
slide.
sir. Yes, sir.
Make it in display mode, presentation mode.
Is it okay, sir?
Yeah, but it has to be in presentation mode. Towards the right hand bottom corner, there is one icon. You click that icon. Presentation. Just before the volume. Just before the display percentage.
And go to slideshow here.
Slideshow. Go to slideshow. Just above that.
Slideshow, not slide. Slideshow. Look at here. Transition, animation.
Are you seeing these icons, small icons at the bottom right corner of your computer, of your screen?
Ask him to go to presentation mode.
No, she doesn't know presentation mode, I suppose. Yeah, that one. Yes, click that. Yeah. Go to your first slide and start.
Good afternoon, sir. So, my oral presentation topic it is optimization of zolmitriptan nanostructured lipid carriers in the management of migraine disease through nose to brain delivery. So, first of all, what is the migraine? It is the third primary cause of neurological health loss worldwide. So this is it is mainly related to the episodes of moderate to severe unilateral headaches. It is also occasionally accompanied by the discomfort and heightened sensitivity to the light and sound. So to overcome all these problems, so the triptan family drug we have used, that is the zolmitriptan.
Hello. Are you online, Miss Snail? Miss Snail, are you online? I think she has lost the connection. My goodness. Are you still adjusting? Hmm.
Okay, sir, sorry, due to technical issue. So zolmitriptan, this drug we have used to overcome the problem of migraine disease. So this triptan family drug, they are the agonist of 5-HT1B1D receptors that are frequently used to treat the migraine headaches. So it is the tryptamine derivative that constricts the blood vessels in the brain to relieve this migraine disease. But this zolmitriptan, it is having the disadvantages like poor permeability, poor bioavailability. So it is also shows some effects like nausea, partial pain, elevation. So due to that, we are going to make the nanostructure lipid carriers of this zolmitriptan drug to overcome these problems like low permeability, low bioavailability. So the main work, it is aimed at the present work, it is aimed to develop the optimized zolmitriptan loaded NLC for the first time by using the box Bankan design of experiment approach. To the best of our understanding, this zolmitriptan analysis, they are given from nose to brain delivery to improve the permeability with the QBD approach, which is not yet reported. So the main novelty of my work, that is here the QBD approach by using this box Bankan DOE method, it is not yet reported. This is the main novelty. After that, the methodology, in that first the screening of solid lipids were done. Here we have used the various solid lipids like stearic acid, presidol, ATO5, comritol, triplet ATO, gelisil, and comritol HD5 ATO. From among all that, we have selected the solid lipid, that is comritol HD5 ATO, because the drug, it is freely soluble in that solid lipid. And screening of liquid lipid, here we have used the different oils as a liquid lipid, that are cinnamon oil, olive oil, hazelnut oil, and castor oil. Here, from all that, the castor oil, it is more in that the drug, it is more soluble, so we have selected this castor oil. After that, this preparation of analysis by using the zolmitriptan. Here the three steps are there. In step 1, the solid lipid, it is melted at the temperature higher 10 degrees Celsius of that than that of its melting point. Then the castor oil containing zolmitriptan, it is added into that solid lipid with the continuous shaking by using the magnetic stirrer. After that, the lipid phase containing this zolmitriptan, it is in that we are going to add the aqueous phase, which is containing the surfactant to improve the emulsification of this particular preparation. Then this preparation, it is subjected to the probe sonicator to make our analysis of zolmitriptan by using this probe sonicator. So this is the hot melt. This whole process, it is known as the hot melt emulsification ultrasonication. So optimization of this zolmitriptan by using the box bank and QBD approach. This box bank and QBD approach, it is mainly containing the 3 factors and 3 levels. So which are the 3 factors we are going to use here? The independent variables, there are 3 independent variables we have used, and 3 dependent variables. The 3 independent variables, that are the content of total lipids, surfactant concentration, sonication time. And dependent variables are the particle size, polydispersibility index, and entrapment efficiency. So these are, here we have made the 12 batches of this analysis containing zolmitriptan. And these are the 3 levels, that are low, medium, and high. After that, characterization by here, the particle size, dispersibility index, and zeta potential. Here the form F1, F2 up to F12 batches we have prepared. The particle size of each batches here we have shown. Then polydispersibility index here we have shown, and entrapment efficiency, which should be near to 100%. And here in next table, that is the drug loading capacity. And last one, that is the F13 batch, which is the optimized batch, which is having a zeta potential, that is minus 22.4 millivolt, which shows it is having a electrostatic stability. Our formulation, it is having a electrostatic stability. After that, here the particle size of optimized NLC and zeta potential of NLC, we have here we have shown the graph of this particle size and zeta potential. Here the zeta potential, it is found minus 22.4 millivolt, which indicates the electrostatic stability. After that, XRD study, the XRD study, it is mainly shows the crystallinity of the particular this zolmitriptan analysis. Here the sharp peak, which is here we got in each of the graph, that shows the crystalline nature of that particular analysis. After that, DSC study, diffraction scanning colorimetry study. Here, the sharp endothermic peak shows that the crystalline nature of the drug, and DSC of this thermogram of optimized zolmitriptan analysis batch indicates the successful entrapment of zolmitriptan in the analysis. So, here this DSC mainly shows the entrapment of the zolmitriptan inside the analysis. After that, optimization by this QBD Box-Benken method. Here, the response of surface contour plots we have got, which exhibits the determination coefficients ranging from 0.9147 up to 0.9476. The insignificant lack of fit, which is more than 0.05, confirming the good predictive reliability. So this is the optimized determinations of this particular zolmitriptan analysis. Then the counter and overlay plot. The yellow region represents the design space where all the criteria were satisfied. The gray zones indicates the constraint violation. The optimal formulation correspondent to the lipid concentration, which is having 1600 milligram.
Excuse me, Miss Snell, I will have to interrupt you because we are falling short of time. Your time is over now. Okay. I may hand over the presentation to the respected judges for cross questioning.
Sir, after that, only in vitro study, in vivo study.
Yeah, we would have allowed if the conclusion slide would have been in process. Okay. So whatever you have presented, the judges may ask you further. Over to you, sir. Sir, you are muted.
Well, you just conclude your.
Yes, sir. So this analysis, here the main novelty of this my research work, that is, we have used the Box-Benken QBD approach to optimize the most stable and effective formulation. Then multi-response plots reveals that the design window where optimized level and surfactant concentration else. So particles, we got the particle size, minimum particle size, that is less than 200 nanometer. Then high polydispersibility index, that is more than 92%. The low polydispersibility index, which is less than that of the 0.3, which should be less than 1. Then the entrapment efficiency, it is high, that is more than that of the 92%. The desirability, it is more than 0.8. The probe sonication, which approach was used to produce the lowest particle size, polydispersibility index, and greatest percentage of the entrapment efficiency. So this is the main conclusion of my work.
From my side, the question is, how migraine disease? What is migraine exactly?
Sir, it is
actually you have shown migraine disease. So how is it disease? Is it disease?
Yes, sir.
How?
Or disease.
Migraine occurs
because headache, nausea, all these symptoms.
Diseases. You yourself have mentioned afterwards in the second slide. Okay. Second question is, are you presenting first time?
No, sir.
You are not mentioned anywhere slide number.
Sorry, sir.
Okay, what is there is no question of sorry. We are learning, you are learning from this. What is the hydrodynamic diameter? You have mentioned somewhere hydrodynamic diameter. What is it? Who did this work?
Yes, here hydrogen.
Who did
258.
work?
That is the party.
You only know? Okay. What is hydrodynamic?
So it shows the particle size, hydrodynamic diameter, that is between 100 to 1000 nanometer. And the peak shows that it is up to 258.6 nanometer. That is hydrodynamic diameter.
Okay, sir.
So your dosage form was nasal spray, right?
Yes, sir.
So tell me, what are the in vitro study you have done? In vitro study for the nasal spray?
In vitro. So here the diffusion cell it is used.
For the formula, so for the formulation, what are the in vitro study in generally one should do? Any idea? I think it is something like spray pattern, plume geometry, something like that. Have you heard something like this? So what was your strength here? What was the strength here? Hello?
Did you get a question?
Yes, sir. Yes.
What was your strength? What was the label claim? You know, because zolbitaftan alone is available in nasal spray. What you have done, you have made
Yes,
it
yes.
close to when. What is your strength? What is the label claim? You know,
Sorry, sir. Sorry.
and what is the actual dosage?
Okay, over to
Sorry, sir.
Vivekanand sir.
Yes, sir.
Yes,
Yes, sir. If the judges allow me, I
माझं
may shift
लॅपटॉपमध्ये ना
to the other candidate.
ऑडिओ नाहीये
I may thank
माझ्या लॅपटॉपमध्ये काय
Nehal
माहिती
Nagpure
त्याच्यामुळे
for
मला
our
इकडे यावं लागतं
presentation.
Thank
And
you, sir.
म्हणजे सगळं मला ऐकू येते पण मी बोलू शकत नाही आणि मी नाही बोलू शकत.
Dr. Basarkar.
I think Dr. Basarkar's microphone is on.
Okay.
Okay.
Thank
So
you. Thank you, sir.
should we, yeah, best of luck for your future
Thank you, sir. I will find
presentations.
out the answers of all these questions which
Yeah,
sir have asked me. Thank you.
that is the real meaning of presentation, defending your presentations, defending your findings.
Yes, sir.
Okay, we may
Yes, sir.
shift
Thank you.
up
Thank
to
you.
next candidate. Mr. Aditya Dongre, I hope he is available. Mr. Aditya Dongre.
Yes, sir.
Okay.
Hello.
Okay. Mr. Aditya, you put your slide, put your presentation. Ms. Nair, would you please remove your presentation? Yes. Okay. Make it in the presentation mode. Slide show.
Okay. Yes, sir. Visible, sir?
Yes. Respected judges, is it visible?
Yes, yes, yes.
Yes, it is visible.
Where is Aditya?
Would you put
Yes, sir,
yourself
this
on camera?
camera not working, sir. Well,
Not
sir,
working. Okay, very
yes,
nice.
sir. Sorry, sir. Sir, I try. I will try.
Okay.
Can I start? Can I start, sir?
Yeah.
May.
Good,
Please start.
good afternoon, respected chairperson and judges. I am Aditya Dogre, first year young farm student from the Department of Quality Assurance, Trinity College of Pharmacy. Today I am here present my work from design to degradation, a green and sustainable HPTLC method for naloxone hydrochloride. So these are the contents. Rationale of a work. Drug profile. The presentation outlines the scientific rationale and drug profile QBD-based method development, optimization and validation, post-degradation characterization, followed by the greenness and blueness assessment, concluding remarks. Uh, Rationale of a work. The comprehensive literature survey revealed that the absence of stability indicating the HPTLC method for naloxone hydrochloride developed using quality by design approach. Furthermore, previously reported analytical method lacked sustainability evolution such as greenness blueness assessment, which are increasingly important environmentally and responsible and economical viable analytical practices. Therefore, the objective present in work to develop and validate a selective stability indicating a sustainable HPTLC method compliant with ICH guidelines and current stress testing. So these are the drug profile of naloxone hydrochloride. Uh, Naloxone hydrochloride is a potent opioid antagonist which used clinically reverse opioid overdose. It possesses the molecular formula C19H22NO4HCl with molecular weight 368 gram per mole. The drug is crystalline white powder, freshly soluble in methanol and sparingly soluble in ethanol. Exhibit a melting point between 200 to 205 degrees Celsius. The determination analytical wavelength. The UV absorption spectrum of naloxone hydrochloride at 20 microgram per ml was recorded. The drug exhibit the maximum absorbance 284 nanometer. Next, the box-Bacon design for the method development. Box-Bacon experiment design was employed systematically study the effect of critical method parameter, namely the mobile phase composition, saturation time, and solvent front distance on the chromatographic response. These parameters were investigated at three different levels, understand their individual and combined influence on the retardation factor of naloxone hydrochloride which selected in critical analytical attribute. The structure approach, the minimized trial and error, experimentation, enhanced method, understand and ensured a final method is a robust and reproducible compliant with a regulatory expectations. Next, we have the ANOVA results. The experiment data obtained from the box-Bacon design statistically using analysis in ANOVA determination significance. This ANOVA result confirmed that the quadratic model was statistically significant, indicating a strong correlation between the selected critical method parameters and the response. The RF value were not due to the random error. Additionally, the lack of fit non-significant conforming model adequately experimental data readily used for prediction and optimization. So there is the formula uh, here. A, B, and C represent the effect of mobile phase composition, saturation time, and solvent front distance respectively. And the linear terms A, B, and C indicate the individual effect of each parameter on RF, while the interaction A, B, AC, and BC shows that how two factors together influence the response. The quadratic terms A square, B square explains the curving, the identifying the optimal condition rather than just linear trends. Then the response surface analysis. The three-dimensional response surface plot illustrate the interactive effect of a method variable on the retardation factor. These plots were instrumental identifying the design space and selecting optimized chromatographic conditions. The 3D response plots represent the optimized predict design identified by the box-Bacon model, in which here we see the red dots within the design indicating the selected conditions are robust and reliable and experimentally achievable. Optimization of HPTLC. Here the mobile phase we use ethyl acetate, methanol, glacial acetic acid, solvent 6:4:0.1 volume by 6 detection wavelength 284 nanometer, retardation factor 54, application volume 2.5, solvent current 80 micrometer. Next we have the method validation. The optimized method was validated according to the ICH Q2 guidelines. The method demonstrates the satisfactory linearity, accuracy, precision.
Hello.
Aditya, go ahead.
I think he has lost his connection. Yeah, he's moving his cursor. Mr. Aditya. Mr. Aditya, are you getting my voice? Hello, are you getting me? Am I audible, respected judges? Amritkar sir, am I audible?
Yes, sir. Yes, sir.
Uh, Mr. Aditya Dongre has lost his connection, I suppose. Or there is some glitch in his connection that he is not able to communicate.
Hello?
His slides are moving. Yeah, you were muted. You were mute for a long time now.
Yeah, Aditya, start.
Audible, sir?
Yeah, speak out. You have got 2 minutes.
Okay, sir. Okay. Sir, this is the greenness assessment. The environmental impact method was evaluated using the AGRI and GAPI assisted tools. AGRI stands for Analytical Greenness Metric Approach. It is a comprehensive tool to use the overall greenness and the blueness assessment. This index is used for economic sustainability of a method. So the conclusion, the greenness, sustainability and stability indicating the HPTLC method and naloxone hydrochloride was successfully developed using the QBD based approach. This method validates the environmentally responsible, suitable routine quality control stability. Thank you, sir.
Okay. What is the use of naloxone hydrochloride?
Is a opioid antagonist, sir.
Opioid antagonist. What
Okay,
is it
sir.
chemically, polar or non-polar?
Sir, please.
What is it chemically, polar, non-polar, mid-polar?
Sir, it is a semi-polar, sir, because it is soluble in ethanol but not in methanol.
Okay, semi-polar. Then how did you choose this solvent mobile phase?
Sir, this is my senior's work.
This is your senior's work. You are only
Yes,
present.
sir. Yes,
Very
sir.
nice.
Yes.
I'm very happy for your honest answer.
Okay.
Very good. That's why there is no point to ask further questions.
So any formulation, any formulation study you have done for this in tablet dosage form, naloxone is available in tablet dosage form, right? So whether you are doing any perform any this validation study by using this your HPTLC method.
Sir, this is by QBD, quality by design.
Okay, check sir.
Okay, slide number 13.
Slide.
13.
Sir, this is the Agri and GAPI studies.
What is it? How did you do that?
Sir, these are the.
Okay, Aditya, go to Vivekanand sir.
Okay. Thank you, Mr. Aditya Dongre.
Thank you, sir.
It is a very, we may say, unacceptable practice or a plague that has plagued the research practices in our country, that a senior's work is presented by a junior. We set up a very wrong practice. Actually, the work which has been conducted by a person in the laboratory has to be presented by himself,
Sorry,
or
sir. Sorry.
the presenter should take the responsibility of whatever is being presented. Okay,
Okay,
it
sir.
is, these are very early days for you, Mr. Aditya, I suppose.
Yes, sir.
So
First year M.Pharm.
from next time you should be taking care of.
Okay, sir. Thank
Okay.
you.
We shift to another candidate as per the list. Miss Nidhi Patel, are you available? Miss Nidhi Patel, this is the last call for you. Okay.
No,
We
sir.
may call Miss Sonali Patil. Is she
Yes, sir. I'm available.
Okay. This is the
Uh,
final candidate, last candidate presenting her presentation, her findings. Please
shall
share
I?
your presentation.
Yes, sir.
Now, Sonali Patil.
Yes, sir. Just a
This is the
moment.
last candidate, sir.
My screen is visible.
Yes.
Shall I start?
Yes. Camera on karo. Miss Patil.
Put yourself on and start the presentation.
Wait a moment, sir.
Camera on कौन present करते?
Uh, am I visible?
No, not it.
No.
A moment. Actually, I have started my video on, but I don't know why it is.
Okay, start your presentation.
Maybe something I need to update in my properties. Shall I start like that, sir?
Yes. You may.
Thank you, sir. My slides are visible, right?
Yes, but make on presentation mode.
Okay.
Okay.
Okay. Good afternoon, all. Myself, Sonali Patil. Today, I am presenting work on QBD approach-based stability indicating method development and validation of fulvestrant with structural characterization of degradants. So fulvestrant, during my presentation, I'm covering the drug profile, then just a brief review of literature survey, aim and objective, then experimental work, validation data, and finally conclusion. So fulvestrant, here are some physicochemical characteristics of fulvestrant here I have mentioned. So it is a white crystalline powder. Its molecular weight is 606, and it is soluble in methanol, ethanol, and insoluble in water. Its pK is 10.32. This fulvestrant drug is actually available in the form of injection form. So it is given by intramuscular route. It is categories estrogen receptor antagonist, and it is used in the treatment of hormone receptor breast cancer. So fulvestrant binds to the estrogen receptor of cancer cell and inhibits the further growth of the cancer cell. So thus this way it acts during the cancer treatment. So now, based on the literature survey, there are few HPLC methods with LSV method are reported. Some bioanalytical methods are reported for fulvestrant. Then few in rat bioanalytical methods in rat plasma are also reported. Then by using stability indicating method by UV are also reported. And whatever the methods are reported are having either high mobile phase, high flow rate, then complex mobile phase, gradient methods. These are the reported for fulvestrant. But force degradation methods are reported, but further characterization of impurities is not reported. And QBD with DOE approach for the development of a stability indicating HPLC method is not reported. So, and there is also further extension of stability indicating method to the by LCMS also not reported. So the aim and objective of this experimental work is to develop a stability indicating HPLC method for fulvestrant using QBD approach, then validate the method, force degradation study and LCMS study. So based on the initial trials of method, the mobile phase chosen was methanol and 0.1% formic acid. So for this experimental work, before starting, we have taken the UV spectra, and the wavelength observed was 225 nanometer. HPLC of JASCO was used with UV and detector. And the column used was HiQ-CC18 254.6 mm 5 micron. So based on the initial trials, this mobile phase was chosen, methanol 0.1% formic acid composition. And based on this composition, this further QBD approach was applied to it. So central composite design approach was used, and design expert version 13 software was used. So for this QBD approach, two factors were used: this mobile phase composition percentage and flow rate. So these are the major two factors which will affect the analytes retention time, its area response. So these two factors are critical. That's why these two factors were selected, and the variables were kept as low, medium, high. So 60-40 for low level, 60-40 composition was used. For medium, 70-30. For high level, 80-20 was used. The flow rate kept chosen was 0.91 and 1.1. So based on these two factors and these three levels, the software has given suggested 9 trials. So these are the data of these 9 trials where the 60, 70, 80% methanol and flow rate from 0.9 to 1.1 was selected, and the retention for each trial, the respective retention time, telling factor and theoretical plates were measured as a response against these factors. So this is the 3D surface plot of fulvestrant using this CCD design to check the combined effect of percentage of methanol and flow rate on the retention time. So from this plot, we can see that as the percentage of methanol increases, the retention time decreases, as the flow rate also increases, retention time decreases. So it is majorly this both factors are affecting the majorly on the retention time. So next, there is not major much impact on the telling factor of these two factors, but we can say that the telling factor is varied between the 1 to 1.6. This is the 3D plot for the theoretical plates. Also, for theoretical plot, we observed maximum theoretical plates for this when for 70% methyl composition was there. So based on these trials, we put this all the 9 trials data, and from that we put in software, design expert software, and we were expecting less retention time than the number of theoretical plates around more than the 4,000. So this software suggested given a solution that for this whatever our desirable results, the 75% methanol 1 ml flow rate, this solution has suggested, and as this desirability is close to 1, so we have selected finalized method based on this solution. So the optimized chromatographic condition are like this. Column is high Q C18 to 54.65 micron, mobile phase methanol 0.1% formic acid composition 75 to 25, 1 ml flow rate, 2 to 5 nanometer wavelength, 20 microliters sampler volume, and the column temperature kept was ambient. So these are the final system suitability for the pulverized by using this optimized chromatographic method. So we got a retention time around 4.6, and the plates and symmetry also found desirable. So once this method is optimized, further this method was validated by using ICH22R1. And during validation, all the validation parameters like specificity, precision, linearity, robustness, accuracy, LOD, LOQ, range, all these were covered. So during specificity, we observed that this method is specific, no interference observed at the retention time of pulverized. Also peak purity was passing. Then for precision study, intraday and interday precision studies were performed. So for intraday, the 6 samples were
Sorry to
prepared.
interrupt, Ms. Patil, your time is over.
If it is your conclusion slide.
Okay, so just give me 2 minutes.
Just go to the conclusion.
Yes, sir. So this degradation study performed, and there we observed some degradants, and these degradants were.
Sorry, Ms. Patil, I will have to interrupt you.
Okay, I will go directly to conclusion, sir.
Yeah, directly go to the conclusion slide, so that
Yes,
the judges may. Okay. Over to you, respected judges. Amritkar sir and Katkadwa sir.
Okay, Miss Sonali.
Yeah.
Who made this presentation?
Yes, sir.
Who made this presentation?
Me only, sir.
But then somewhere you have mentioned slide number. That font is different. In some cases, there is no slide number. In another case, the slide number with the blue color. How is it happen?
Honestly, sir.
Honestly, now you have to be very honest.
No, means I got this is not my finalized version, I will say.
Yeah.
Again, I wanted to make it more presentable manner. Sorry for that, sir.
How do you come to know that 225 is the wavelength for this? If you see the structure, how it has a 225 then?
UV spectra for that.
Chemistry department or physics?
Chemistry, sir. I got UV maxima at 225. I have taken spectra for that.
If you see the structure, how it can be 225? This is just my question. Because we are chemists, I'm looking at a chemist point of view. There is a phenyl ring, aromatic ring is there. We have another substituent. Okay.
Okay.
And what are theoretical plates you have mentioned in some slide number 4?
Slide number 4.
Which slide number 4? I don't know, but somewhere it is there. Slide number 4.
Ha, sir. You can see my screen, sir.
Yeah.
Ha, sir.
Theoretical plates in this HPLC. Okay, over to sir.
So have you quantified LOD and LOQ by this finalized method, LOD, LOQ?
Yes, sir.
So you didn't mention there, right?
It is there in validation, summary of validation parameters, sir.
Okay. And whether you are able to quantify the impurities which you have done, if you see alkaline degradation, oxide degradation, percentage are very less. So whether you identified what are those impurities, additional peaks?
Further, sir, LCMS study done. And in this LCMS study, I got this degradant peak also. So based on the
known impurities? These are the known impurities or unknown impurities?
Sir, with the I have tried to find out the structural degradation pathway, sir.
Whether in pharmacology they mention RT, RRT, something like that for this unknown impurities? RT.
No, sir. Here not mentioned in this chromatogram. But we can see here this LCMS spectra are there, no?
Can you explain that LCMS spectra?
Yes, sir. This is the standard Fulvestrant standards spectra. So here we got this and this mass spectra. So here around in 607, this is the that we got the Fulvestrant peak. So this is the major peak of Fulvestrant. Here is a plain Fulvestrant. So its molecular weight is 6 not say 8. So we got. Hello.
What about the other peaks which have been observed here?
Here, sir, what? For this standard, I will say majorly. Once this our analyte enters in the.
Okay. Over to Vivekanand, sir.
Yes, sir. I would like to add that it being a steroid, the general nanometer in UV spectra, UV range goes up to 350, 390 nanometers. But when we use HPLC, the detectors of HPLC, maybe in older HPLCs, the detectors were only allowed 2 or a couple of nanometers to measure the absorbance. And the literature the candidate has referred seems to be of that end of HPLC, not of pure chemistry or UV absorption. That is why there is the confusion. Okay, so next time you take note to properly refer the literature, Miss Sonali.
Yes, sir. Sure, sir.
Okay, all the best for your future endeavors.
Thank you, sir.
I may declare that all the presentations, all the candidates who have submitted the abstracts and were given a chance to present in the conference are now over, and the judges, respected judges, Dr. Sunil Amritkar and Mr. Katkutwar sir, are requested to tabulate the data collected, prepare the mark sheet and declare the results. You will get some time to tabulate the things, to decide for the winners. There are three winners to be declared by the end of the valedictory session, which is followed, which will start in few seconds. Okay. By the end of the valedictory function, I appeal to our judges to be ready with the results and declare it on your own.
So Chatpati sir will take 5 minutes and then we'll update you, right?
Should we declare internet or after valedictory?
No, you can declare it.
Right now, no?
Yeah,
Okay.
I would
All
like
the
my technical
presenter-
team. I would appeal my technical team to take over the dais and start with the validity function.
Should we declare in a validity or before right now?
Yeah, we will start.
Sir,
You take your
validatory
time for.
function, please
You take your time and convey me when the results are ready.
Our results are ready. When should we declare?
Sir, I request you to declare. Sir, Amrutkar sir,
Yes,
I
sir.
request you to declare after valedictory function, which will be starting very soon. It will take just 5 to 10 minutes.
Okay.
Okay.
So
On
should
my
we
behalf, I would, on behalf of SNJP Pharmacon 25, the scientific committee, I would like to thank the judges and all the participants. All the best for your future endeavors.
Sir, I would like to
Thank you,
talk
sir.
1 minute because all the presenter tried their level best to present their work. Some did a really good job. The research was good. In somewhere they tried their level best to present. In some cases they were a little bit confused, but all have tried a very level best. So I'm very much thankful to all presenter and very much thankful to SNGB for giving us opportunity to evaluate these 9 research articles by presenting by different PhD scholars and teachers. Thank you. Over to sir.
So overall, all the presentation were excellent, but there is some little here and there. So we have taken decision and we decided three prizes that we will declare soon. So I'm very much thankful to SNJP College of Pharmacy, the management, Upasani sir, Basarkar sir, and all the staff of the SNJP College. Yes, Pharmacon is a wonderful event whereby you come to know all the acquisition, what is the update in the recent pharma field. It is a proud moment that Charanjit Singh is associated with the SNJP from last 9 years. So there is a lot of scope in the pharma. Being an industry person, I'm telling you, every year Indian pharma is growing at about 9% growth rate. There is a scope in the area of ANDAs, NCEs. So moving to the further, it is not only in the research, but there is a scope in the area of intellectual property management, in the regulatory sale, in the QBD sale. So the topic which was there in the Pharmacon was the stability indicating methods. So the topic was very excellent. You all know what happened in the recent issue of the cough syrup. What happened? It was only stability indicating method of the propylene glycol, because propylene glycol, there were a lot of cases whereby children were, because of the propylene glycol being used in the cough syrup and the issue of impurity, which was the diethylene glycol, around 25 children were lost. So the topic which was finalized was very excellent. So I'm very much thankful to all the management and everyone as far as this session is concerned. Thank you, everyone, and thank you all the students and management staff. Over to Chatpal, you are, sir.
Should we start, sir?
Yes, sir.
So, respected chief guest of today's valedictory function, Mr. Ashok R. Katkurwar, respected dignitaries, esteemed speakers, session chairs, dear faculty members, researchers, delegates, and my dear students. A very good afternoon to all. On behalf of SSDJ family, I, Dr. Vandana Daga, gives me immense pleasure and a sense of fulfillment to present the valedictory address of the online 2 days virtual national conference SNJB Pharmacon 2025 on practical and regulatory considerations for stability indicating methods in assays. As we come to know the conclusion of this national conference, I am honored to share this moment with all of you. This conference, organized by Shriman Suresh Dada Jain College of Pharmacy, has been a platform for sharing knowledge, discussing challenges, and advancing our understanding of how these methods contribute to the safety, efficacy, and regulatory compliance of pharmaceutical products. I take this opportunity to express my deep sense of gratitude to our esteemed chief guest for the valedictory function, Mr. Ashok R. Katkurwar sir, from the Department of Pharmaceutical Sciences, Nagpur University, Nagpur. Sir brings with him an outstanding blend of over 35 years of rich industrial experience, holding more than 50 patents on his credit, and has significantly contributed to the development and commercialization of various solid dosage forms for India, raw, European, and US markets. His remarkable work in respiratory dosage forms, NDA filing in nasal spray formulations, analysis, technology transfer, and successful execution of numerous industrial projects is truly inspirational for both academia and industry. We are deeply honored by your presence and guidance, sir. So now I would like to request our respected chief guest, Mr. Ashok R. Katkurwar, sir, to enlighten the audience. Over to you, sir.
Thank you, Vandana. Thanks to the management of SNJP Pharmacy College. Thanks, Principal Upasani sir. Thanks, Ganesh Basarkar and all faculty members. It is my pleasure to be part of this event. Pharmacon 225 is a wonderful platform being organized from last 9 years, which helps all academicians and budding pharmacists to know the recent update in the pharmaceutical field. It is a proud moment for all of us that we have got a mentor, Dr. Saranjit Singh, a great scientist and a true academician in the pharma field for our guidance. The 2-day program was excellent, especially the presentation delivered is so good. As an industry person, you know, we have got India is having largest US FDA approved plants. 1/3 of the drugs which are being imported in US are being manufactured in India. There is a lot of scope in the ANDA, US generics. So there is a scope in the area of complex generics, transdermal delivery, osmotic delivery, liposome, NC, and PK studies. So from the career perspective, what I feel is not only production R&D, there's an area which is neglected nowadays. Is we have to look into the intellectual property, development quality assurance, regulatory affairs. So let us have a more industry and college coordination in the days to come. Coming back to the symposium, the topic was excellent practical and regulatory consideration for stability indicating method assay. Because it is not only the assay 95 to 105, what is most important is your impurities. Because getting assay 95 to 105 doesn't carry any meaning. What are the impurities? What are the LODQ? What is the your quantification and detection limits? What are your toxic effects? Because a single complaint will damage your company's record. So the topic which was declared or which was finalized by the SNJP Pharma is a very excellent, which I feel. So once again, I thank all the management of SNJP, Upasani sir, management staff, Ganesh Basarkar, all my colleagues and all the participants who were there in the today's symposium and today's seminar. Thank you all. Thank you all.
Thank you, sir. Over the past 2 days, this conference has truly served as an excellent academic platform for the exchange of ideas, knowledge, and innovations in the field of pharmaceutical sciences. The enthusiastic participation of academicians, industrial professionals, research scholars, and students from across the country has made this event a grand success. I would like to extend my sincere gratitude to our distinguished keynote speakers, Dr. Saranjit Singh, former director, NIPER, Chandigarh, Dr. Prasad Thakur Desai, Chief Scientific Officer and Vice President, Indus Biotech Limited, Pune, Dr. C.R. Patil, Professor IPR Shirpur, Dr. Prashant Murumkar, Assistant Professor from Maharaja Sayajirao University of Baroda, and Dr. Vishal Zambre, Associate Professor and HOD of Pharmacochemistry Department, PCEIS School of Pharmacy, Pimpri Chinchwad University, Pune. Your expertise and insightful presentations have not only broadened our knowledge, but have inspired us all to continue pursuing excellence in our respective fields. I would also like to sincerely thank all our distinguished resource persons whose expert lectures, technical insights, and real-world perspectives have greatly enriched this conference. Now, I request Dr. G.D. Basarkar, Coordinator of SNJB Pharmacon 2025, for his concluding remarks. Over to you, sir.
Thank you, Dr. Vandana Daga. Respected Chief Guest of today's valedictory function of SNJB Pharmacon 25, Mr. Ashok Katkurwar, former Senior General Manager, Research and Development, Landmark Pharmaceuticals Limited, Nashik. Speakers, esteemed delegates, faculty members, research scholars, and my dear students. As we come to the conclusion of SNJB Pharmacon 25, I feel a deep sense of pride and gratitude. Over the course of this conference, we have witnessed enriching scientific deliberations, very much stimulating paper presentations, as mentioned by our respected judges, and meaningful interactions that were truly reflected the spirit of research innovations. So on behalf of organizing committee, I sincerely thank our respected Honorable Chief Patterns, mentor, and technical evaluation experts, Dr. Sunil Amrutkar sir and Ashok Katkurwar sir, for their constant guidance and encouragement. I extend my heartfelt thanks to our management, administrative officer, and our principal sir, Dr. Chandrashekhar Upasani sir, for their unwavering support and trust, without which this conference would not have been possible. I would like to acknowledge the enthusiastic participation of research scholars, faculty members, and students for various institutions. Your active involvement, curiosity, and scholarly contributions that were truly made SNJB Pharmacon 25 a vibrant and memorable academic event. As we conclude, I am confident that the ideas exchanged and the knowledge shared during this conference will definitely translate into meaningful research outcomes and future partnerships. Let us carry forward that knowledge gained here and continue to work together for the advancement of pharmaceutical education as well as in research in India. I once again thank each one of you for being a part of SNJB Pharmacon, for making it a grand success. I look forward to meeting you again at future academic gathering at SNJB Pharmacon 26. Till then, bye bye. Thank you very much. Over to Vandana Daga.
It's now time to recognize the outstanding contributions made by our participants in this conference. We received several excellent presentations, each reflecting the high level of expertise and passion for pharmaceutical research. The prize distribution is a moment to celebrate the hard work and dedication shown by our participants. It is not just about winning, but also about encouraging excellence, fostering innovation, and inspiring others to continue their journey in pharmaceutical sciences. We are delighted to announce the winners of the best presentations. So now I request Dr. Amrutkar sir and Katkurwar sir to please announce prizes for today's presentations. Over to you, sir.
Thank you, Vandana. Myself and Dr. Amrutkar, we have discussed together and we have made few decisions, right? So as per the presentations are all very good, but now we have to decide 3 prizes, right? So third prize is Snehal Nagpure. The winner for the third prize is Snehal Nagpure. Okay.
Daga madam.
Yes, sir.
Third prize to Snehal Nagpure.
Yes, sir. We are listening.
Second prize goes to.
Second prize goes to Varun Joshi.
Varun Joshi for second.
Joshi.
Okay.
Any guesses for the first prize?
Sonali Patil, I suppose.
So first prize goes to Vinesh Kumar.
Okay.
Congratulations. Those who are the winners will be communicated through their mail addresses. Thank you.
Thank you.
Congratulations, all the winners. However, I will also like to extend my appreciation to everyone who participated in this conference. Your contributions, questions, and enthusiasm have played a key role in making this event a success. Each one of you has contributed to the rich tapestry of knowledge shared during these 2 days. Congratulates all. Now it's time. It's my honor to deliver the vote of thanks for the virtual national conference on practical and regulatory considerations for stability indicating methods in assays. This event has been an incredible success, and we owe it to combined efforts of many individuals and teams. Firstly, I would like to express my deepest gratitude to our distinguished speakers. Their contributions have truly made this conference a remarkable one. Their expertise in stability indicating methods, along with their ability to explain complex regulatory and practical considerations, has left a lasting impact on all of us. A special thank to the management and organizing committee headed by Dr. C.D. Upasani, Dr. G.D. Basarkar, and the rest of SNJB Pharmacon team for their relentless efforts in ensuring the smooth execution of this virtual conference. The planning, coordination, and dedication have been outstanding. We also extend our appreciation to the technical team who ensured that we were able to connect seamlessly despite the challenges posted by the virtual format. Without their support, this conference wouldn't have been possible. Thank you to all the participants for your active engagement and valuable contributions. The questions, discussions, and feedback shared throughout the sessions have enhanced the quality of this conference. Now, as we wrap up, let us take with us not only the knowledge gained, but also the connections we have made with fellow professionals. Let us continue to engage with each other, collaborate on research, and push the boundaries of what's possible in pharmaceutical sciences. With that, I officially declare the virtual national conference on practical and regulatory considerations for stability indicating methods in assays is closed. Stay connected, stay inspired, and wish you continued success. Thank you. Thank you all.
Thank you, sir. Amritkar sir, Katkuwar sir.
Thank you, sir. Thank you, sir.
Thank you.
Okay, thank you, sir, for giving opportunity.
Okay, bye bye.
Thank you, sir. Bye bye.